Good morning. Good morning, one and all. On the occasion of 102nd birth anniversary celebration of our founder chairman, Shri Padma Bhushan, Dr. B.V. Raju Garu, we have a tribute from Vishnu Dental Institution wishing him a 102nd birth anniversary.
And Sri Venkata Narasimharaju Garu were blessed with a child. He was none other than Dr. Bhupati Raju Vissam Raju Garu. His enthusiasm to prove himself with an unending quest for higher education made his mother Srimati Buchi Rajeshwari Garu to pledge her gold to finance his educational journey from his hometown to Banaras Hindu University and graduate in chemical technology in the year 1941. He was also awarded with a management degree from the Harvard University of the United States of America. While everyone judged him to be an educationist, destiny had other plans for this gentleman. His childhood habit of working hard and his perseverance made him excel in each and every job that he undertook. He proved himself in the field of cement industry and carved a remarkable niche for himself. Eventually, he was made the chairman and managing director of the Cement Corporation of India. Under his dynamic leadership, cement sector witnessed enormous growth in India and made him to be fondly remembered as the doyen of the Indian cement industry. In recognition to his efforts in uplifting the cement industry, in 1977, the Government of India had conferred him with the prestigious Padma Shri Award. Then he started Rasi Cements in 1979 and went on establishing many industries such as VISHNU Cements, Rasi Refractories, Rasi Ceramics, and many more. In his lifetime, he established 35 industries and provided livelihood to many families. He also received an honorary doctorate from the Jawaharlal Nehru Technological University for his vigorous performance in the cement industry. After his retirement, he decided to do something for his homeland. He believed that education would be the key tool to bestow upon the children at his place to bring a major change in their lives. Hence, the seed to establish educational institutions was sown, which grew up to be the tree called Sri Vishnu Educational Society. Since inception, the Sri Vishnu Educational Society has been continuing to provide qualitative yet affordable education to empower thousands of children and their families. A journey of 1,000 miles starts with a single step. Many people had their own apprehensions about starting educational institutions within rural areas. But his mountainous zeal made Dr. B.V. Raju Garu go on and turn his belief, if your thoughts are good, then your deeds will also be good, into an acceptable reality by transforming, inspiring, and nurturing lives through education. He believed education and social service were the efficient tools for social uplift. Towards this endeavor, he adopted many schools in the surrounding areas of his birthplace and transformed them into the happiest places of learning. His ancestral property was converted into Dr. B.V. Raju Knowledge Center to facilitate education for all in the remote areas. He made sure that no area was untouched. He also adopted leprosy patients and underprivileged people who were being ignored during the times in society and changed their lives by providing good quality healthcare, housing infrastructural facilities through Dr. B.V. Raju Foundation. His relentless efforts had converted bare lands into the most exciting educational institutions at Bhimavaram, Andhra Pradesh and Narsapur, Telangana. Dr. B.V. Raju Garu was passionately driven by his belief, serving to mankind is serving to God. His sense of social responsibility was an endless battle. In recognition to his remarkable services to the society, he was bestowed with the Padma Bhushan Award by the Government of India in the year 2001. The great personality and the social visionary left this world in the year 2002 at the age of 82.
After a great tribute to our chairman, Dr. B.V. Raju Garu, today we mark the third day of Vishnu Adhyaan. Welcome you all once again. I hope you all are benefited by last 2 days lectures, series of presentations. Before we proceed, I introduce myself. I myself, Dr. Sukrija, Senior Lecturer from the Department of Oral Pathology, will be the host for today's session. Coming to our first speaker, now I take a privilege to introduce our first speaker of the day, Dr. Swagatika Panda ma'am, who is a professor from the Department of Oral and Maxillofacial Pathology at Siksha O Sandan Dental College, Bhubaneswar. Ma'am has got a strong interest in research. She has an interest in pathogenesis and facts of diagnostic procedures in oral cancer. Her research interests, which motivated her to undertake initiative for early diagnosis of oral cancer with the aid of chairside techniques through DBT and BIRAC sanctioned projects. To her credit, there are 64 quality publications in several medical, dental journals, including high impact factor journals such as scientific reports, oral oncology. Ma'am has applied for 2 patents and 1 copyright. Ma'am is going to enlighten us today on the topic basics of immunity and hypersensitivity. I request all the participants to post your queries in the chat box. They will be answered subsequently at the end of the lecture. I now hand over to you, ma'am. Session is all yours.
A very good morning to one and all. As a part of VISHNU ADHYAAN 2022, VISHNU Dental College has brought us all here in this prestigious virtual platform to discuss about the basics of basic medical science. I am Dr. Swagatika. We all are here today to discuss basics of immunity and hypersensitivity, which is a very important subject in medical science. There are so many reasons why we need to study immunology, but for the time being, our focus is to understand the concepts of immunology so that we will be able to reproduce that knowledge during exam, on the exam papers. And you must be knowing that there are so many important long questions and short notes coming from this chapter. So we will be discussing under these headings with a brief introduction on immune system and immunity. We will discuss types, components, and phases of each immune system. Afterwards, we will discuss hypersensitivity, in which we are going to discuss mechanism of each type of hypersensitivity. So this last pandemic has taught all of us what is immunity. Yes, it is the body's defense mechanism against invading pathogens. It could be virus, bacteria, fungus, parasite, or it could be our own dead and denuded cell or tumor cells. The immune system is not an organ system. It is the lymphoid organ which gives rise to this functional system, that is immunity. And this lymphoid organ is of 2 types: primary lymphoid organ. Thymus and bone marrow are the primary lymphoid organs, and this is the site where cells of the immune system originate and mature. Then the secondary lymphoid organ, that includes tonsils, lymphatic vessels, spleen, lymph nodes, etc., where lymphocytes gets activated and differentiated. And most importantly, the cells of adaptive immune system, they recognize pathogen here in the secondary lymphoid organ. Immune system can be categorized into 2, innate and adaptive. Innate is what we are born with, and adaptive is acquired immunity, which we acquire during the development of our body. The differences between innate immunity and adaptive immunity are: innate immunity are they manifest very fast, and they cannot distinguish between types of pathogens. It cannot distinguish whether the external invader is a bacteria or virus or fungus. It cannot distinguish, and it cannot remember also the last encounter with which pathogen it has encountered last. It cannot remember. Whereas adaptive immunity is a little bit late to respond, and it is antigen specific. It mounts specific response for bacteria, fungus, parasite, and virus. Even it recognizes whether it is gram negative and or gram positive, and accordingly it responds. And most important aspect of adaptive immunity is development of memory cells. As a part of immunity, certain memory cells are generated and they are freely circulated in the body. When the next encounter happens, the response is even stronger and massive. Starting with the innate immune system, this has got 4 defense mechanisms: physical, chemical, microbiological, and biological. Biological has got 2 components: cellular component and humoral component. Physical defense mechanism happens due to the skin and mucous membrane. Chemical defense mechanism happens due to the secretion of physical defense mechanism. For example, sweat, which is a secretion of skin, prevents the growth of microorganisms because of the antimicrobial peptides in it. Similarly, hydrochloric acid released from stomach mucous membrane prevents the growth of microorganisms. And another example is lysozyme, which is present in saliva and tear, that prevents the growth of microorganisms. The third defense mechanism, that is microbiological, that is happening because of natural gut and oral microbiome, which are our normal commensals of our body. They also prevent and inhibit the growth of other pathogenic microorganisms. The fourth and most important defense mechanism of innate immune system is biological barrier, which includes cellular component and humoral component. Cellular components are WBC, white blood cells, otherwise known as leukocytes, and humoral, that is present in serum. They are 3 C's, you can remember like that: complement, coagulation, and cytokine. One by one, we will discuss, starting with cellular components of innate immunity. There are so many types of leukocytes: monocyte, eosinophil, basophil, neutrophil, and lymphocyte. Eosinophil target helminths as they release certain molecules which cause pores on their surface and lysis of the parasites. And this is also involved in allergic response. Basophil is another group of cell which is also involved in allergic response. Then monocyte, that's a very important cell in innate immunity. Monocyte and neutrophil, both of them are responsible for phagocytosis. We'll learn what is phagocytosis. And lymphocyte, a variant of lymphocyte is large granular lymphocyte, otherwise known as NK cells or natural killer cells. They are born to kill cells, to kill infected cells. So one by one, we'll see how each of them is functioning. The role of neutrophils is at most important in innate immunity because they phagocytose. How do they phagocytose? The first step is the microbe adheres to the plasma membrane of neutrophil. Then it is being ingested by the help of pseudopodia of neutrophils, and then it is ingested into the cytoplasm of the neutrophil as phagosome. That phagosome fuses with a normal cellular organelle of neutrophil, that is lysosome, and thus phagolysosome is formed, which releases so many digestive enzymes and the microbes are digested. And when it is partially digested, there are certain residual bodies and they discharge through the process of exocytosis. And it comes out from the neutrophil cytoplasm. In this process, there is rapid release of reactive oxygen species, superoxide anion and hydrogen peroxide. This is known as oxidative burst or respiratory burst, which kills internalized pathogens. There are, other than neutrophil, there are another 3 phagocytic cells. They are monocyte, macrophage, and dendritic cells. Monocyte, when it migrates, macrophage, and dendritic cells, they are located which encounters external antigen. To all the phagocytic cells, apart from being phagocytic, all of them are known as antigen presenting cells because they present the antigen to the immune cell, which is going to defend it, these monocyte, macrophage, and dendritic cell, they are going to present the antigen. They phagocytose, that means they engulf the pathogen, and the degraded product they are going to present to the other immune cells. That is why they are known as APC or antigen presenting cells. And the last common feature is the release, all of them release cytokine. So a small note on dendritic cell, as I mentioned, they are located on the epithelium of oral mucous membrane and epidermis of skin, because they are the organ which first encountered with the external injurious agent. So when they encounter the virus or bacteria, they capture those dendritic cells, they capture with the help of their dendritic processes, they capture the pathogen, and then they migrate to lymph node. So this is a picture of dendritic cell. You can appreciate how with the help of dendritic processes, it is engulfing the pathogen, and then it is engulfing it into its cytoplasm, where the pathogen is broken down. Those pathogen breakdown products are again, they come up to the cell membrane of the dendritic cells, and later on it is presented to the appropriate immune cells. A group of cells are lymphocytes, which is again 3 types, B cell and T cell. Those 2 types are involved in adaptive immunity. So we'll discuss that later in this presentation. And the natural killer cell, otherwise known as large granular lymphocyte, that is a lymphocyte which takes part in innate immunity. As the name is suggesting, it naturally kills, but not all the cells. It differentiates. It has got the potential to differentiate between healthy cell and infected cell. When the cell is infected, it recognizes and it releases those cytotoxic granules which contain porphyrins and proteases, so that those enzymes destroy the infected cell. So we have finished discussing the cellular components of innate immune system. Next, we are going to discuss the humoral components. Those are present in the serum. They are your complement proteins, coagulation proteins, and cytokines. Let's see how this complement protein mediates innate immunity. This complement protein is a group of distinct plasma proteins ranging from C1 to C9, and their job is specific. So those complement proteins, they perform this immunity function of immunity by 3 mechanisms. The first mechanism is formation of membrane associated complex, and that is undertaken by C5 to C9 complement proteins. When there is any pathogen, the C5 to C9, those group of proteins, they recognize the pathogen and come and gets attached to the cell membrane of the pathogen. And there a pore is being created on the cell membrane of the bacteria or virus, the cell membrane of the pathogen. And because of that pore, through that pore, extracellular fluid passes into the bacterial cytoplasm. And that is how there is imbalance and the bacteria cell gets lysed or gets ruptured and it is dead. The second mechanism is through opsonization mediated phagocytosis. Opsonization, the literal meaning is coating. So basically C3b, that is the complement which is involved in this process. It coats the bacterium. This is your, you can see the C3 complement protein, which is cleaved into a larger part and a smaller part. The larger part is C3b and the smaller part is C3a. The larger part, what it does, it coats the bacterium. It coats the bacterium and prepares, as you marinate the chicken and then prepare it for cooking. So similarly, C3b, that is a complement that opsonizes or marinates the bacterium so that it can be recognized by the phagocytes like macrophage. And simultaneously, what happens, C3A, the role of C3A is it recruits macrophages to the site of immune response. So that more and more number of macrophages come, they recognize C3B coated bacterium and they can engulf or they can phagocyte those. The third mechanism is mediated by three complement proteins. They are C4A, C3A, and C5B. They release anaphylatoxin, which activates mast cells to release histamine, and it leads to inflammation. The second protein is humoral protein is coagulation protein. So in our body, in normal homeostasis, there is a balance between coagulation protein and anticoagulant. But when the balance is disrupted because of increased number of pathogens and increased number of cytokine release, the coagulation proteins are synthesized more and more in number, so that inflammation is accelerated and apoptosis is facilitated. So this is how coagulation protein takes part in innate immunity. The third component of humoral mediated innate immunity is cytokine. Those are small proteins. They are secreted by cells of the immune system. They can take their action by autocrine method and paracrine method. Autocrine is the cytokine which is released from the cell. It again influences, it again activates the same cell from which it is secreted to undergo proliferation. Whereas the paracrine is, it influences the adjacent neighboring cell to undergo proliferation. So a cytokine has got both autocrine and paracrine behavior. This is a very important player in both innate and acquired immunity. Three most important cytokine, we should have a basic knowledge regarding that. They are interferons, interleukins, and tumor necrosis factor. We'll just fast, we will see what is the role of these cytokine. First is interference. It is written usually written as IFN. It is released by the monocyte or lymphocyte, which is mostly virally infected. The virus which infects monocyte and lymphocyte, those cells, they release interferon and they contribute to the immunity in three possible mechanisms. In first mechanism, what do? They warn the uninfected adjacent cell to destroy RNA and reduce protein synthesis so that the virus cannot replicate in those cells. The second mechanism is it also warns the adjacent infected cell to undergo apoptosis. And the third mechanism is this interferon also activates macrophages and lymphocytes so that they can phagocytose the pathogens. The second cytokine is interleukin, which is released mostly by lymphocytes and macrophages. And they have three important mechanisms of action. First is they help in proliferation of immune cells. Then the second is they activate plasma cells and B cells so that antibody production is increased. And the third mechanism is they activate the immune cell, specifically cytotoxic T lymphocytes and macrophages so that phagocytosis takes place. The third cytokine is tumor necrosis factor, TNF, which is released by macrophages that activates dendritic cells or antigen presenting cells, which in turn release certain other cytokines that activates T cells into T helper 1 cells and T helper 17 cells. Both of them take part in inflammation. So after finishing the components of innate immunity, we are moving towards phases of innate immunity. There are 4 steps in which innate immunity happens. First is PRR and PAMP recognition. PRR has to recognize PAMP, PAMP has to recognize PRR. Then the phagocytosis of the cell is happening. Then rest of the immune cells in the neighborhood, they become alert. And fourth is there is a coordination between the innate immunity and the adaptive immunity. explaining step by step. The first step is PRR and PAM recognition. So what is PAM? That is pathogen associated molecular pattern. That means it is present on the microbes. And PRR is pattern recognition receptors, that is present on the host cells. So the host cell, the host immune cell has to recognize the pathogen. That is how PRR, the brightest example of PRR is toll-like receptor. You can see TLR2, TLR5, TLR2, and they are very specific. They recognize their respective specific PAMs. PAMs for TLR2 is lipopeptide. PAM for TLR5 is flagellin. PAM for TLR2 is peptidoglycan. So you can see there are specific PAMs for specific TLR. So the differences between PRR and PAM, because these are very new terminologies, I would like to again brush up. That pattern recognition receptors are transmembrane proteins present on macrophages and few other immune cells that recognize and bind to PAM, which are present on microbes. The brightest example is toll-like receptors, and PAM are surface characteristics of microbes which are recognized by PRR. For example of pathogen-associated molecular patterns are lipoteichoic acid that are specifically present in gram-positive bacteria, lipopolysaccharide, which is present in gram-negative bacteria that makes gram-negative bacteria unique, and flagellin, etc. So after the first step, second step is white blood cell, mostly your neutrophils and macrophages, they throw their pseudopodia around the bacteria and they engulf the bacteria and they bring it to their cytoplasm. That is how the bacteria is digested inside the cell. This process is called as phagocytosis. And in the step 3, after it has been phagocytosed, the lysosome inside the phagocytic cells, they release certain cytokines, certain chemicals, which warns the neighboring cell. It kind of sounds the alarm and warns the adjacent cell that the pathogen has already invaded, so you better be careful. And the last step is there is a link established between the innate immunity and adaptive immunity by the dendritic cell. The dendritic cell initially was immature or naive dendritic cell, which is present in the epithelium of oral mucosa and epidermis of skin. When it is engulfing the pathogen, then it gets activated and then it migrates to lymph node where it activates the naive T cells that later we'll discuss in detail. So this is the summary. Any invading microbe has to be encountered by external defenses of innate immunity at the outset. So after that, the internal defenses like our cellular component, humoral component of innate immunity, they come into action. And after that, acquired immunity come into action. So this is the transition from innate to adaptive immunity when the dendritic cells, they engulf the pathogen and travel to lymph nodes. Their maturation of key players of adaptive immunity takes place. Key players of adaptive immunity are T cells and B cells. This is a beautiful picture showing cells involved in innate immune system, cells involved in adaptive immune system, and you can see the dendritic cells and large granular lymphocytes. They are the connecting players between innate immune system and adaptive immune system. So next we are going to discuss adaptive immune system, which is classified broadly into two, active and passive. Active, which we develop ourselves, whereas passive, which we acquire from someone or something. When the immunity is acquired from mother to baby through breastfeeding mostly, that is called as passive natural immunity. And when when the vaccination is given to acquire immunity, that is called as passive artificial immunity. Active immunity is further categorized into two: cell-mediated and humoral mediated. Humoral mediated is otherwise known as antibody mediated. One by one, we are going to discuss. Cell-mediated immunity, as the name is suggesting, the cells are involved in this defense mechanism. So what are the cells? First, as I said, antigen-presenting cell, mostly dendritic cell, has to reach to the lymph node, and then the antigen, that is the degraded product of the pathogen, has to come to the cell surface. And it has to be served, it has to be offered to the immune cell, to the naive immune cell. Here it is naive T cell, by the help of T cell receptor. So see, there are three things: MSC protein, which are present on antigen presenting cell. They act like serving plate. They offer the antigen to the naive T cell through the T cell receptor present on T cell membrane. So this naive T cell becomes activated and differentiate into helper T cells and cytotoxic T cells. This activated T cell that also secretes one interleukin, that is interleukin 2, which activates NK cell, natural killer cell. And this interleukin 2 has got an autocrine action on the T cells which has secreted IL. This T cell also secretes another group of cytokine, so that cytotoxic T cell can proliferate further. That process is known as clonal expansion of cytotoxic T cells. And the cytotoxic T cell and NK cell together kill infected and altered cell. Rest of the cytotoxic T cell remain in the circulation as memory cell. As memory T cell. So this is the gist of cell-mediated immunity. We heard a new term, that is major histocompatibility protein. In short, it is called as MSC protein. So let me brief what is MSC protein. They bind peptide fragments derived from pathogen, which we call it as antigen, and display them on the cell surface so that it can be easily recognized by appropriate T cell. There are two types of MSC protein, MSC1 and MSC2. MSC1 is typically present antigen to CD8 T lymphocytes. That they present antigen to cytotoxic T lymphocytes only, whereas MSC2 present its antigen to CD4 T lymphocytes. MSC1 typically is found on surfaces of all the nucleated cells in our body, whereas MSC2 is exclusively found on surfaces of antigen presenting cells and activated T cells. Then coming to humoral mediated, otherwise known as antibody mediated immunity. So the same situation, antigen presenting cell has already migrated to lymph node, where it has already activated the T cell with the help of MSC protein and T cell receptor. This T cell, when it gets activated, it also activates, it releases certain cytokines and activates the naive B cell. When the naive B cell proliferates, it leads to plasma cell differentiation, and part of B cells goes into the circulation as memory B cell. This plasma cell, the very important function is it secretes antibody. Antibody has got several regions, and the antigen binding site of antibody recognizes the epitopes on antigen, and thus antigen-antibody complex is formed. How is that antigen-antibody complex is offering the immunity? By two mechanisms. Either it neutralizes the antigen directly by blocking the antigen binding site, or it induces the recruitment of other immune cells to promote antigen removal or destruction of the antigen. antigen. So these are the two mechanisms by which antigen-antibody complex offers immunity, and this is known as humoral mediated or antibody mediated immunity. So summary of T cell immunity and antibody immunity, very important. You should know that T cells, they recognize infected cells. The cells are already infected. T cells, they recognize those infected cells and kill those infected cells. But in antibody immunity, the white blood cells or B cells or plasma cells, they release antibody and they prevent the cell to be infected. So this is the difference between T cell immunity or cell mediated and antibody mediated immunity. Next, coming to phases of adaptive immune response. Adaptive immune response takes place in several steps. So we will see what are the steps. Next, we will see phases of acquired immunity. In the very first step, the antigen needs to be recognized by the immune cells of secondary lymphoid organs. Here, your naive T cells and B cells must recognize the antigen-specific receptor molecules on their surface and become activated. This is called as recognition phase or clonal selection. Afterwards, in the next step, in order to mount a stronger immune response, there need to be many a number of activated T cell and B cell. So in the next step, it proliferates. The activated immune cells proliferate. This is called as clonal expansion. And in the next step, the effector cells, they neutralize or destroy the antigen, which is known as effector response. And once the effect is established, once the immunity is established and the pathogen is about to decrease, the antigenic effect is about to decrease, there occurs declining phase where the number of proliferating activated immune cells starts declining. It is also called as clonal decline, clonal deletion. And during this process, few B cells and T cells remain in the body as memory cell, so that in the next antigenic encounter, they can exert a stronger and massive response. So these are the phases of acquired immunity. These are the differences between humoral or antibody mediated and cell mediated immunity. The site of activity for humoral is in the extracellular fluid, whereas in the cell mediated, the site is location of antigen presenting tissue like skin or mucous membrane. And the cell type involved in humoral mediated immunity is B cell, because that is what is going to produce antibodies. And in cell mediated, it is the T cell which is taking part. Then humoral mediated immunity is very fast, whereas cell mediated immunity is delayed and slow. Antigen in humoral mediated immunity is extracellular mostly, and in cell mediated immunity, the antigen is mostly intracellular pathogen and cancer cells. So MSC protein which is involved in humoral mediated immunity is MSC class 2 proteins, and in cell mediated immunity, these are class 1 protein. You must know these differences. These are the basic differences between the two types of adaptive immune system. As of now, we have discussed so many terms which probably we are not very thorough with. So let's recapitulate the terminologies. Let's brush our terminologies. The most common term which we come across while studying immunology is antigen, otherwise known as immunogen, which is the substance that elicits an immune response. It may be pathogen, it may be any injurious agent, anything, but that has to elicit immune response to be called as an antigen. And hapten is a substance which itself cannot elicit any immune response, so it lacks antigenicity. But when it combines with any carrier molecule, then it forms a complete antigen which can induce an immune response. Then whenever we speak about antigen, definitely we are going to speak about antibody. Antibodies are produced by plasma cells. They're essential parts of adaptive immunity, and they bind specifically to a unique site on the antigen, which is known as epitope. They possess antigen binding site known as FAB. That is the region, that is the site which is bound to epitope of antigen, and it is also called as immunoglobulin. The size is approximately 10 nanometer. This is how an antibody looks like, having two heavy chain, two light chain. Both they are identical, those heavy chains and light chains. And it has so many region. It has two region, basically constant region and variable region. And it has got antigen binding fragment and constant fragment. These are the things you can brush up just before the exam. So there are the 5 types of immunoglobulins present in our body. M, D, E, A, G. The characteristic feature of each immunoglobulin you can read from this slide. So this is what is immunity in short. Although it is a very complicated process, but the basic is this much, that when the pathogen or any foreign object enters into our body by breaking the continuity of our skin or mucous membrane, it is first captured by immature dendritic cells. They are located in the skin epidermis and oral mucosa epithelium. They recognize, yes, it is a foreign agent, it is a pathogen, and then they capture the pathogen, and then they migrate to the lymph node. Along with the dendritic cells, there are neutrophils also. So these are the left side of the picture presents the innate immunity, that we are born with that kind of immunity. Then when the dendritic cell migrates to lymph node, there it activates the naive T cell. Naive means the immature T cell, that is activated to a mature T cell and mature B cell. B cell secretes antibodies, which is a very important component of humoral mediated immunity, is a part of adaptive immunity. And this activated T cell also also differentiates another T lymphocyte, that is cytotoxic T lymphocyte or CD8 plus T lymphocyte, that destroys the infected cells. And that is through cell-mediated immunity component of adaptive immunity. This is the summary of immunity. And these are the functions of immune cells. In case any short note comes on functions of immune cells, you can refer to this slide and you can draw a diagram similar to this so that it will be more mark-fetching. So this is what is immunity, and when you relate the immunity in our day-to-day life, you can find that people in our life are like antigens with different targets, different virulence. So in first exposure, we deal with our innate immunity, but in second exposure, we deal with our acquired immunity, the knowledge of handling different kind of people. So simply, they just elicit our potent immune response. There's a beautiful quote related to immunity and our life. So the next topic of discussion is hypersensitivity. As the name is suggesting, you must be assuming that hyper means exaggerated and sensitivity is response. So exaggerated tissue response is what hypersensitivity. No, it is wrong. Hypersensitivity is actually tissue damaging immune reaction. Do remember like this, hypersensitivity is tissue damaging immune reaction. Johansson has defined hypersensitivity like it causes objectively reproducible symptoms or signs initiated by exposure to a defined stimulus at a dose tolerated by normal subjects. It is elicited by antigens, which could be exogenous or endogenous, and it involves immunological or non-immunological mechanism. Non-immunological mechanism is known as pseudo-allergy or idiosyncratic reaction. There are 4 types of hypersensitivity reaction. Type 1 is known as immediate. Type 2 is known as cytotoxic or antibody dependent. Type 3 is immune complex, and type 4 is delayed type hypersensitivity. One by one, we are going to discuss all. Type 1 hypersensitivity, you can memorize by remembering this. You can memorize this sentence and you will be able to write it in the exam paper. One antibody named immunoglobulin E caused mast cell to release histamine. It has got this type 1 hypersensitivity is executed in two phases, sensitization phase and reaction phase. Sensitization phase is when the antigen, there is first exposure of the antigen presenting cell, that is dendritic cell, to the antigen, and thus antigen presenting cell gets activated and Th helper T cells are formed, which in turn activates B cells, and that in turn is differentiated into plasma cells which secrete antibodies, specifically immunoglobulin E, that goes and gets attached to the mast cell surface, and thus degranulation of mast cell and basophils happen. Histamine, leukotrienes, and other mediators are released. Whereas reaction phase is the re-exposure phase. That means when the antigen is encountered in the body for the second time, then it is directly recognized by immunoglobulin E that are already present on the mast cell surface, and degranulation happens and the reaction response is a little faster. So this is sensitization phase and reaction phase of type 1 hypersensitivity, which is the most common type, and it is provoked by re-exposure to a specific type of antigen known as allergen. Then the type 2 hypersensitivity, you can memorize all the features of this type 2 hypersensitivity by remembering this sentence: two autoantibodies attacking me, IgM and IgG. It takes almost 2 to 24 hours to develop, and there are two antibodies named as IgG and IgM. They bind to cell surface antigen. Antigen is already fixed on the cell surface. Immunoglobulin G and immunoglobulin M, which are free, they come and gets attached to the cell surface antigen. This is how antigen-antibody complexes are being formed, which destroys target tissue by three mechanisms. What are those three mechanisms? Activated complement system activation, opsonization and phagocytosis, antibody-dependent cellular cytotoxicity. So this type 2 hypersensitivity, it is being executed by three mechanisms. The first mechanism is complement system activation. What happens when the antigen on the cell surface is reacting with the antibody, then this complement system gets activated, specifically the C3A and C5A. And we have already discussed the formation of membrane-associated complex, which inserts into the cell membrane of the particular cell. When the target cell is RBC, then there occurs formation of pores on the cell surface of RBC. That is how extracellular fluid comes into the cell, and there is osmotic imbalance, because of which RBC hemolysis happens. The second type is opsonization mediated type 2. Opsonization, we have already discussed that it is a form of coating the bacterium. So here, when the antigen on the target cell interacts with the antibody, then macrophages, they, with the help of complement proteins, when the complement proteins opsonize or coats the pathogen, macrophages, they recognize those coated particles. Those coated pathogen are recognized by macrophages, and slowly they engulf the pathogen. Because it has been recognized easily, they can engulf it easily, and phagocytosis happens. The third mechanism is antibody-dependent cellular cytotoxicity, where antibody IgG comes into play. Antigen on the cell surface gets attached to the freely circulated IgG, and then their natural killer cell comes into play. Natural killer cell, it recognizes the IgG and antigen complex by the help of Fc receptor, present on its surface, and then it straight away kills. So then coming to type 3 hypersensitivity reaction. Type 3 hypersensitivity reaction we can memorize as 3's complex get the hint as it involves the complement. So what happens? IgG and IgM, both the antibodies take part in this type 3 hypersensitivity. They are freely available in the circulation. Similarly, soluble antigens are also freely available in the circulation. So both are free. Antigens are free, antibodies are free in the circulation. Inside the blood vessels, this is the scenario. The soluble antigen reacts with the antibodies like IgM and IgG, and as immune complex is being formed. That immune complex goes and gets deposited on the tissue surface. Once it gets deposited on the surface of the tissue, like synovium of joints, kidney glomerulus, or alveolar surface of the lungs, then it activates one complement, that is C1. That in turn activates neutrophil, and neutrophil that gets activated and that release granules which are loaded with oxygen reactive species, and it destroys the cells on the tissue. So the examples are systemic lupus erythematosus, serum sickness, and reactive arthritis. Then coming to type 4 reactions, you can remember this sentence: 4 T cells like to rave, but the response is more delayed. This is otherwise known as DTH, delayed type hypersensitivity. Similar to type 1, it also has got 2 phases, sensitization phase and effector phase. In sensitization phase, antigen presenting cells, those are your dendritic cells, they activate CD4 helper cells. CD4 helper cells again gets differentiated into multiple T helper cell subsets like T helper 1, T helper 17, and it also gets differentiated, though in a small amount. Cytotoxic T cells are also activated from this CD4 helper cell. Then in the effector phase, what happens? The sensitized T helper cell, which are already formed in the first phase, they release certain cytokine, most importantly interferon gamma, that activates the rest of the resting macrophage to turn into or to transform into activated macrophage. Then the class 2 MSC molecule on the activated macrophages become activated, and that is how phagocytosis takes place. So what happens? Generally, pathogens in effector phase are killed rapidly with little tissue damage. But if this delayed type hypersensitivity is prolonged, then it can develop into visible granulomatous reaction. This is how your granulomatous inflammation is developed. So in summary, there are 4 types of hypersensitivity reaction: type 1, 2, 3, and 4. Type 1, 2, and 3 are antibody mediated, whereas type 4 is cell mediated. Type 1 is where antigen is free and foreign to the body, and antibody is immunoglobulin E and it is fixed on mast cell. Examples are anaphylaxis, asthma, angioedema, and allergic rhinitis. Type 2, where antigen is fixed and intrinsic, antibody is free. And antibodies, the name of the antibodies are specifically IgG and IgM are involved. You can see this example, how the antigens are fixed on the red blood cells and antibodies are free. When the reaction takes place, it leads to destruction of RBCs, and this is a pathogenesis of autoimmune hemolytic anemia, which is a very important example of type 2 hypersensitivity reaction. Another example is erythroblastosis fetalis. Type 3 hypersensitivity, in that antigen is free, antibody is free. As I showed you in the lumen of the blood vessel, both antigen and antibody are free and they form immune complexes which are deposited on the tissue surfaces and destroys tissue. Examples are systemic lupus erythematosus, reactive arthritis, serum sickness. Then coming to the last type 4 hypersensitivity, you won't find any antibody here because it is cell mediated. Which are the cells? It is the antigen presenting cell which activates T lymphocyte. The example is tuberculosis, chronic transplant rejection. This is the applied aspect of immunology. There are several diseases that those are caused by aberrant immune responses, and this is beyond the scope of the present slide. But however, while writing long question, you may add this applied aspect on immunology, which can fetch you good marks. These are my references, and I'm very grateful to Vishnu Dental College, particularly Dr. Ravikant, for giving me this opportunity in this prestigious platform. Thank you very much. This is a learning experience for me too. Thank you very much.
Thank you, ma'am, for your interesting and interactive session. Ma'am, am I audible?
Yes, you are very much audible. Thank you so much. I hope PG students can gather at least the basic concept of this chapter.
Yeah, ma'am. Ma'am, we have few queries from the students. Can I ask you, ma'am?
Yes, sure.
They have asked, what is the declining phase in humoral immunity?
Declining phase in humoral immunity is the cell and the antibody, when the quantity of the antibody is being decreased. That is what is declining phase.
Ma'am, we have another question, ma'am.
Yes.
Oral manifestation of full food allergy syndrome.
Yes.
Yeah.
See, this is the question, right?
Yes, ma'am.
There are a spectrum of oral diseases which are the manifestations of, what did you say, allergic disorders, right?
Allergic syndrome. Yes, ma'am.
Yes, the most common is we know that medicamentosa, and so many drugs are there which cause local allergic reaction or systemic allergic reaction. Local allergic reactions are manifested as white patches, ulcerations, and you must have heard of this oral lichenoid reaction, which is a very similar presentation. It overlaps, the features are overlapping with lichen planus, oral lichen planus. And you diagnose, that should be your provisional diagnosis, first provisional diagnosis. You discontinue the medication which you suspect it could be the possible reason, and then you can see that it has healed. Otherwise, you can go for histopathology, where you can differentiate between lichen planus and lichenoid reactions.
Thank you, ma'am.
That's altogether different chapter. So we can discuss that in a longer, like in a larger, when we have sufficient time. So probably there are hardly 5 minutes left.
Okay, ma'am. Okay, ma'am. Thank you, ma'am. That was very clear and crisp lecture, ma'am. Ma'am, we have a certificate of appreciation from Vishnu Dental College, ma'am.
Yes.
Adhyaan. Please accept, ma'am.
Yes. Thank you so much. The certificate is beautifully designed. Thank you. Could you please take little time to mail this certificate?
We'll mail you the certificates, ma'am.
Yes, thank you.
Thank you, ma'am. Thank you so much.
Okay, bye.
Moving on to the second presentation of the day, we have Coming on to the second presentation of the day, we have the microbial culture and culture media to the oral microbiologist, Mr. Anil Babu, MSc, medical microbiologist, graduate from the Kasturba Medical College, Manipal, Udupi, Karnataka. Currently, he is pursuing PhD program from Meenakshi Medical College, Kanchipuram, faculty in the Department of Microbiology at VISHNU Dental College, Bhimavaram. He is expertised in automated blood culture system, immunology like ANA Global, PCANA, serology, bacteriology, and bacteriological examination of water, evaluation of efficacy of disinfectants. So now the presentation will be continued by Dr. Anil. Over to you, sir.
Good morning, everyone. And this is Anil Babu S. and Department of Microbiology, VISHNU Dental College, Bhimavaram. And today I will be going to talk about microbial culture, media, cultural methods. So we will see in this session definitions of culture media, classifications of culture media, culture methods, anaerobic culture methods, quality assurance, significance in dentistry. The common definition of culture media. So it is just like a food for bacteria, and it is term given to microorganisms that are cultivated in laboratory for the purpose of studying them, microorganisms studying them. Coming to the mediums are certain combination of ingredients that will boost up and growth and supporting for the growth and cultivation of microorganisms outside their natural habitat. Requirements for the growth of bacteria. So for the preparation of culture media, so we need a distilled water. Then coming to the next thing, peptone, the peptone which will be extracted from the golden granular powder and obtained meat soya flour, and they are complex mixture of partially digested protein substances and proteases, examples of amino acids, polypeptides and phosphates and minerals, etc. Riboflavin. Coming to the meat extract, so by the name only you will come to know, and these extracted material extracted from the meat, and it contains the majority of protein degradation products and carbohydrates and inorganic salts. Electrolytes, the electrolytes source mainly from sodium chloride and salts. Agar, and it is acting like solidifying agent and obtained from seaweeds, does not provide any nutrition to grow for the organisms, and it will melt at the temperature of 98 degrees Celsius and solidifies at 42 degrees Celsius. Coming to the classifications, the culture media classifies in many ways that are based on oxygen requirement and physical state and nutritional factors. Coming to the according to oxygen requirement, and it was divided into two groups of microorganisms, aerobic medium and anaerobic medium. According to physical state, there are three types of medium available for the physical state. First one, liquid media. I gave the examples peptone water and nutrient broth, and you will also give glucose broth. And second thing, semi-solid medium, nutrient agar stamps and mannitol motility medium also. And third thing, solid medium. Solid medium, it gives examples of blood agar, nutrient agar, and Muller-Hinton agar. According to nutritional factors, and this is very important thing in culture media, there are four major groups: simple media, complex media, synthetic media, special media. And let's elaborate simple media. So the simple media, for the best example thing, nutrient broth, the preparation of nutrient broth, and it contains peptone water and meat extractions of 1%, and this mixture will form a nutrient broth. And if you add in this mixture to glucose, and it will become glucose broth. And instead of adding glucose, and if you add 2 to 3% of agar into that nutrient broth, and it will become nutrient agar. Complex medium, the complex medium, the best, which means the complex medium other than simple media are complex. And we can give the examples like blood agar and McConaughey's medium and Muller-Hinton agar medium. The complex medium have the one special thing. The complex medium have added ingredients of bringing out their certain properties like hemolysis or non-hemolysis, lactose fermenters or non-lactose fermenters. All these certain properties can be obtained these culture media, which we gave the examples of media. Synthetic media, synthetic media and preparation is prepared from pure chemicals, and it will be useful only in the research aspects and metabolic activities are identifying metabolic activities on bacteria. And this is not at all useful in patient clinical diagnostic purpose. Examples of medium: dubose medium. Coming to special medium, and it is divided into 7 subgroups of medium. The enriched media, followed by enriched media, enrichment media, selective media, differential media, indicator media, transport media, sugar media. The first thing, enriched media, the basal media added with substances, I mean the basal media in the sense nutrient agar. If the nutrient agar added with substances such as either blood or serum or egg, and it is known as enriched medium. And examples of medium: blood agar, chocolate agar, low flux serum sloop, and Lowenstein junction medium. And I put here some pictures regarding to the examples of medium. The functional thing, blood agar, and which facilitates and bringing out their certain properties like hemolysis or non-hemolysis. And you can appreciate in the blood agar medium. And based on that hemolysis, and you can differentiate gram positive coca. And coming to the chocolate agar, it is um, mainly to isolate very delicate organisms like gonococci, meningococci, especially for fastidious microorganisms. Loeffler's serum sloop only for specific organism. Example: Corynebacterium diphtheria. It is useful only for Corynebacterium diphtheria main. And coming last example, Lowenstein junction medium. The Lowenstein junction medium, which it is prepared from it is prepared by egg yolk. And example of organism, Mycobacterium tuberculosis. Second example, enrichment media. The enrichment media means it facilitates the isolation of particular species from a mixed inoculum. I'll discuss one important case history here. For example, if a normal person affected with Vibrio cholerae, for suppose the patient is visited to OPD for the physician. The physician and suggested to collect the stool sample. Further proceedings for identification of Vibrio cholerae. And during collection of stool sample, the sample may get contaminate. The finally the sample it reaches the microbiology lab further findings. And then we have to inoculate the stool sample for alkaline peptone water. I gave the example here, alkaline peptone water. The alkaline peptone water having enrichment substances, and these substances will grow allow only Vibrio cholerae. If any contaminants in the sample, for suppose that contaminants will be inhibited by enrichment substances. So that is the reason enrichment media. Second point, it is liquid medium, and which favors, as we already discussed, favors the multiplication of a particular species. Either and selectively favorite or inhibitory substances that suppresses competitors. Competitors, I mean contaminants. Examples of enrichment media: selenate F broth and tetrathionate broth, alkaline peptone water. Selenate F broth only for isolation of Salmonella and Shigella, and tetrathionate broth for Shigella, alkaline peptone water only for Vibrio cholerae isolations. Selective medium, and it is a solid medium, and the selective medium, it is followed by enrichment media. The only difference, the functional thing is similar to selective media and enrichment media. The only difference is this is solid medium, that is liquid medium. The same, and inhibits the growth of unwanted bacteria but permits the growth of wanted bacteria in a form of colonies. Examples of medium will be used for this selective medium: deoxychelate citrate agar and launched in junction medium, and deoxychelate citrate agar for Salmonella and Shigella also. Differential medium, and these medium contains some substances that will help to distinguish and differing characteristics of a bacteria. It is called a differential medium. Example medium: the medium it's facilitating to differentiate the organisms. Example of medium: Meconchis medium. The Meconchis medium contains peptone, lactose, agar, sodium tercalate, and neutral red. This is mainly for the gram-negative differentiations, and based on the lactose fermentase, we can differentiate gram-negative bacilli. And after inoculating sample onto the medium, Meconchis medium, just keep it for 37 degrees Celsius for 24 hours. After 24 hours, if you see the pink color colonies on the medium, and you can report LF colonies. LF in the sense lactose fermentase colony will appear in the pink color. If you see the colorless colonies, that will resembling to the non-lactose fermenters, and by this way you can differentiate lactose fermenters and non-lactose fermenters. Indicator medium followed by Meconchis medium and blood agar medium. Meconchis medium, as we discussed already, the Meconchis medium facilitates bacterial growth along with lactose fermenters or non-lactose fermenters. The lactose fermenters and non-lactose fermenters is differentiated by color formations. So color is indicating whether it is LF or NLF. Coming to the blood agar medium, blood agar medium also facilitating, and the certain properties is bringing out there hemolysis or non-hemolysis, and based on that, and it will be indicated the organisms. Transport medium, the transport medium only for samples containing delegate organisms such as gonococci, meningococci, and Vibrio cholerae, which they not survive outside the body for the longer period. It need emergency to subculture in the particular medium. Examples of Stewart's medium and buffered glycerol saline medium and Carey Blair medium is, these three mediums is used for the transport medium. The last one, sugar medium, and it is used to determine the ability of an organism to ferment a specific carbohydrate present in the medium. Sugars, examples of sugars: glucose, lactose, sucrose, maltose, mannitol, ribose, ribomanose, dextrose, arabinose, arabinogalactose. There are many types of sugars will be used for the identification of microorganisms. For example, one bacteria capable to ferment all types of sugars, we named something. Second organism can ferment only lactose or sucrose, we named different. The third organism, for example, and it cannot ferment any type of sugar, and we named non-fermenting microorganisms. Based on that sugar fermentations, we are identifying the microorganisms. Culture methods. The culture methods used for growing microorganisms are known as culture methods, and different culture methods are used for the culture of bacteria. Purpose of doing cultures: to isolate bacteria and pure cultures from the mixed inoculum, and demonstration their properties, and obtain sufficient growth of preparation of antigens, and type isolates of methods such as bacteriophages and bacteriocin susceptibility, and determinative sensitivity to antibiotic susceptibility testing, estimate viable counts, and maintain stock cultures. Inoculation of media. And before going to talk about culture methods, and we should know inoculation of media, how to introduce a sample to medium. The sample will be introduced by inoculation loop, and there are two types of inoculating loops: round loop and straight wire loops. Methods of culture: there are 6 types of cultures are cultural methods available for the streaks: streak cultures, lawn culture, stroke culture, stab culture, pour plate culture, liquid cultures. And let's discuss about culture streak cultures. And this is most routinely employed in the clinical microbiology department for the isolation of bacteria in a pure form of any type of specimen. uh, for suppose when we receive the sample, so you just take some amount of sample through the inoculating loop and placed on the medium and make a oval shaped smear. Then immediately you have to do first lines from the two words of uh, inoc from the two words of where primary inoculum. Then loop has to be sterilized in between, then let it be for cool. After cooling, the again you start doing lines from first line to second lines and second line to third lines, continue, and third line to fourth line. From the fourth line you just make a zigzag formation, and keep it for 37 degrees Celsius for the incubator, and 24 hours of overnight incubation. After overnight incubation, so you will get the bacterial colonies along with isolated colonies. The isolated colonies will settle down at the region of zigzag method. Lawn culture, and the very important thing, uses of lawn cultures, and it is used for the antibiotic susceptibility testing in everywhere in clinical microbiology department by using diffusion method or Kirby-Bayer methods. Bacteriophage typings. Bacteriophage typings also can be done in lawn cultures, and obtaining a large amount of bacterial growth of preparation of antigens and vaccines. Example of media used for the antibiotic susceptibility testing: McConkey's medium. Antibiotic susceptibility testing procedure: just take one sterile test tube with distilled water and add some amount of bacterial colony into that mixture, and just put one sterile cotton swab and swab entire the media will be used. McConkey's medium, Muller-Hinton agar medium. Then place antibiotics, different antibiotics on that medium, and keep it for 37 degrees Celsius for the overnight incubations. So after overnight incubations, you can appreciate sensitive pattern and a resistant pattern. So that is the process of antibiotic sensitivity testing. Streak cultures, and it is made in tubes and containing agar slants also. And nutrient agar is most commonly used in research aspects mainly, and also we can use maintaining stock cultures for the studies. Stab cultures. So in this method, the culture is made by puncturing at the center of medium. Applications of stab cultures: demonstration of motility of bacteria, demonstration of gelatin liquefactions, and maintain stock cultures of bacteria. Pour plate method, and the pour plate method only for urine cultures. I kept here some pictures related to the pour plating culture method, and it is mainly employed for urine cultures. So when we receive the urine sample, you just pour some amount of maximum amount, in the sense 10 to 15 ml of urine sample, and spread everywhere on the medium. And after that remaining soapernent sample you have to discard. Then keep it for same period, 24 hours overnight incubations. After 24 hours overnight incubations, if you see the 1000 colonies above for the colony count, and you can report as 10 to the power of 5, I mean 1 lakh colonies are counted on the medium. And if you see the less than 1000 colonies, I mean 871 colonies, 10 to the power of 4, 87 colonies, 10 to the power of 3, 8 colonies, 10 to the power of 2, and less than 8 colonies, 10 to the power of 1. That is probable chance of getting infection of patient. Liquid culture, the liquid culture, it is employed for only blood cultures. Why? Because of blood is all the time it is sterile, and the blood is transferred through the pipettes or syringes also. Anaerobic cultivation method, to isolate anaerobic bacteria and which grow only in the presence of absence of oxygen, but grow in the presence of carbon dioxide like 10% or 20%, and creating anaerobic condition called anaerobiosis, and established by various methods. There are two ways. And first way, anaerobic condition can be created in a airtight closed container. Examples of anaerobic jar, as you know already, and Mecanthos fields jar, and made up of, and these anaerobic jars is made up of either steel or polycarbonate. In another way, anaerobic conditions can be created by certain using anaerobic media in test tubes, and these medias will be reducing the oxygen and creating anaerobiosis conditions. Methods of anaerobiosis: anaerobic gas pack methods, and this is routinely employed in clinical microbiology department, and evacuation and replacement method by displacement and combustion of oxygen. Chemical methods, absorption of oxygen by chemicals. Candle jar method, displacement of oxygen. Anaerobic gas pack method. The gas pack is commercially available in the available by high media, and this method is routinely employed in everywhere in clinical microbiology laboratories. The gas pack is which contains sodium borohydride and cobalt chloride, citric acids, and sodium bicarbonate. And these chemicals generate hydrogen and carbon dioxide. So when water is added about 10 ml, some gas pack do not needed water. Coming to the carbon dioxide, which will boost up for the anaerobic microorganisms. And coming to the hydrogen will combine with oxygen during and keeping inside the inoculating loops. The oxygen will react with the hydrogen, and it will form a water, and it will be settled down at the bottom of the anaerobic jar. And you can also see here the picture of anaerobic jar. So, and before doing it, the plate should be inoculated with samples. So, and before doing this, the inoculating plates, the petri dish plates should be keep inside the chamber, and immediately you have to open the gas pack, commercially available pack, open and keep it there. Along with that, and you have to maintain anaerobic indicator that is also available from the high media. So there is an indicator we kept inside the anaerobic jar, methylene blue, and during anaerobiosis, the methylene blue, it turns the white color. That means the anaerobiosis is present. And after that, immediately close the lid with tightly and keep it for incubation maximum 72 hours. And evacuation replacement method. So for this, so we used to use Mecanthos field jar is used. There are two types, field jar, metal and glass. There are, and you can also see here, there are two lets. So one is outlet, one is inlet. The other one is pressure gauge, and which will showing the concentration of carbon dioxide. And before doing it, your opening outlet and removal of oxygen by vacuum, So after this process, when you finish this process and immediately you close that outlet and open the inlet and passing the first passing of 90% hydrogen gases through the inlet, then after that 10% carbon dioxide will be passed, and that will be creating anaerobiosis. To maintain anaerobic, chemical method, absorption of oxygen by chemicals. There are two types of chemical methods. Okay, pyrogalol. So pyrogalol, in this method, a tube containing pyrogallic acids and sodium hydroxide is placed inside a airtight jar. That absence of oxygen absorbs oxygen and produces anaerobic conditions. The same thing. And chromium and sulfuric acid, these two chemicals react in the presence of oxygen, forming anaerobiosis, producing anaerobiosis. Advantages is and simple, very reliable to use in clinical laboratory. Candle jar method. I hope you all aware on this process. And inoculating plates should be keep inside and close the candle lighted. Also, along with the plates, you should keep it inside and tightly close the lid with tightly. Then automatically carbon dioxide concentration will be produced. That concentration is stimulating the growth of most of the bacteria. Anaerobic medium. There are two important types of anaerobic medium: Robertson's cooked meat broth and thioglycolate broth. Coming to the Robertson's cooked meat broth and cooked meat particles such as beef heart substances. The beef heart, which carries carbohydrate, protein, unsaturated fatty acids. The unsaturated fatty acids absorbs oxygen and creating anaerobiosis. Coming to the carbon dioxide and protein, if protein is utilized by one bacteria, for suppose, and that reaction is called as proteolytic. Example of bacteria, Clostridium tetani. If the meat containing carbohydrate utilized by one special type of organism, for suppose, that reaction is called as saccharolytic. Examples of Clostridium perfringens. And thioglycolate broth and thioglycolate used as a reducing agents of absorption of oxygens. The bacterial growth is identified by turbidity. Now, turbidity might be, you will see in the bottom of the test tube. Coming to the quality assurance, checking whether the process of working are properly or not. This is the indications, quality assurance. Chemical, there are two indicators, chemical indicators and biological indicators. Methylene blue, we already discussed in the gas pack system. And during anaerobiosis, the methylene blue, it turns white color. That means anaerobiosis, it was success. Coming to the biological indicators, and Pseudomonas aeruginosa will grow if anaerobiosis is not maintained. Thank you.
Thank you, sir, for providing us a great expose on the topic. Queries will be answered before moving to the next lecture. Sir, we have few questions from the participants. May I, sir?
Yes.
What are the uses of high chrome agar media?
So especially high chrome agar media used for the differentiate of different candidial species, and this is the main importance in mycology department, mycotic infections. And then coming to this clinical aspect, and it will also use in urine cultures to differentiate urine pathogenic, and it will suppresses the protease growth. Protease is inhibiting other substances that is acting like contaminants.
So we have one more question, sir. Which are the special media for anaerobic organisms?
So special media for the solid, we have neomycin blood agar is most commonly used. The neomycin, it is type of antibiotic associated with the blood agar medium. So we will provide some nutritions to anaerobic microorganism. Then neomycin antibiotics will suppresses the aerobic organisms. If the sample, for suppose, if sample, we receive the sample. So the sample will carry the aerobic organisms and anaerobic organisms. For suppose anaerobic organism is acting like a pathogen. We just concentrate only on a pathogenic organism. So these neomycin antibiotic will suppresses the unwanted substances, unwanted bacteria.
Thank you so much, sir. So we have a certificate of appreciation from Vishnu Adhyaan, sir. Kindly accept.
Thank you, ma'am. Thank you.
Thank you so much, sir. We will move to the third session of the day and introduce our eminent speaker, Dr. Veeravarmal ma'am, who is a professor and head of the department in Rajamuttaya Dental College and Hospital, Annamalai University, in the Department of Oral and Maxillofacial Pathology. We welcome you, ma'am. She has been a topper in first MDS using her study at Mahatma Gandhi Dental Postgraduate Institute of Dental Sciences, Puducherry. She has been awarded with the Best Researcher of the for the academic year 2019. Ma'am is well trained in IHC and worked with more than 80 primary antibodies of different manufacturers and got training for PCR and cell culture techniques also. And she has been an external examiner for various universities in and around the Tamil Nadu and Karnataka. We welcome you, ma'am. Before ma'am begins her topic on the significance of calcium and phosphate metabolism, I request all the participants to post your queries in the chat box regarding the topic. They will be answered subsequently at the end of the lecture. Session is all yours, ma'am.
Good morning to everyone. I'm very glad to communicate with the first year MBS across the country through this platform. For making this opportunity, I would like to thank VISHNU ADHYAAN 2022 team, and also special thanks to Dr. Shweta Ramesh for suggesting my name. The topic in which I'm going to talk is the significance of calcium and phosphate metabolism. Calcium and phosphate plays a very, very important role in multitude of physiological activities. Their rearrangement can result in skeletal-related cardiovascular morbidity, and sometimes it is life-threatening. The calcium and phosphate metabolism is well regulated by different types of hormones by targeting the organs like bone, kidney, and intestine. Calcium is very much essential for a hormone and a neurotransmitter release. For muscle contraction, both the calcium and the phosphorus is needed. Calcium is mainly involved in enzyme activation, a coagulative pathway. The cellular energy is provided by the action of phosphorus. The cell signaling is also involved in the phosphorus. The membrane stabilization by the formation of the phospholipids is with the help of phosphorus. Four hormones are mainly involved in the calcium and phosphate metabolism. They are 1,25-dihydroxycholecalciferol, otherwise called as vitamin D, parathyroid hormone, calcitonin, fibroblast growth factor, number 23. Now, our body, we have a 1 kg of calcium. So 99% is present in both, 1% is present in the soft tissue. 600 grams of phosphorus is present, out of the 80% is present in bone and 50% in the soft tissue. For a well-being, the serum calcium phosphorus, vitamin D, parathyroid hormone, alkaline phosphate level should be maintained. So these are the normal range of all these parameters. Calcium have a major role in a bone mineralization. It is seen in a crystalline form, that is like a calcium hydroxyapatite crystals. A soluble form is seen in serum and intracellular areas. In serum, 50% is an ionized calcium, 41% is a protein bound, only 9% is a non-ionized. There is a balance between the calcium intake and the excretion through the feces and the urine. Per day, 1 gram of calcium is needed, and this 1 gram of calcium is excreted through the feces and urine. So, whatever is absorbed through the GIT will enter into the bloodstream. So, there is a balance between the calcium that is deposited in the bone and the calcium that is entering in the bloodstream through the reabsorption. During the filtration, 9,980 milligrams of calcium is entering into the filtration system. In that, 9,880 is reabsorbed, only 100 milligrams entering into the urine. There is the exchange of a calcium between the cell and the extracellular fluid, that is a blood. So the whole mechanism is well balanced, the intake as well as the output. The phosphate is present in our body either in an organic or an inorganic form. The organic form is seen in the cellular component of the blood. The inorganic form is seen in the serum. The 35% of inorganic is complex with the sodium, calcium, and magnesium. 10% is bound to the protein. The remaining 55% is present in a free form. Similar like a calcium metabolism, the phosphorus intake and the excretion is well balanced. The daily requirement is 1 to 1.5 grams. So the amount of phosphorus that is excreted through the feces is 0.5 gram, through the urine 0.7 gram. Whatever is absorbed through the intestine enter into the intracellular areas as well as the extracellular areas. Whatever the phosphate that is entering into the bloodstream, that is balanced by the deposition of the same amount into the bone. So the balance between the absorption as well as the excretion is well balanced. So if there is going to be a fall in a calcium or a phosphorus level because of some rearrangement or because of some hormonal influence, then the absorption of the calcium and the phosphorus from the intestine will be altered. Either it will be increased or decreased according to the need. The reabsorption from calcium and the phosphorus from a kidney also will be increased or decreased according to the need to maintain the serum calcium and the phosphate level. Vitamin D is one of the main hormones that regulates the calcium and the phosphate metabolism. 7-dehydroxycholecalciferol is a substance present in the subcutaneous tissue on a sun exposure but is converted in a cholecalciferol. This cholecalciferol undergo hydrolyzation in the liver with the help of 25-hydroxylase. The 25 cholecalciferol undergoes further hydrolyzation in the kidney under the influence of an enzyme called as 1-alpha hydrolase. So this 25-hydroxycholecalciferol converted into a calcitrol, that is nothing but 125-dihydroxycholecalciferol. For this convention, two other factors is needed. One is the parathyroid hormone, another one is a fibroblast growth factor, number 23. The main role of vitamin D is mineralization. It helps in the deposition of calcium and the phosphate in the bone matrix. And it also aids in the absorption of the calcium and the phosphate from the intestine. Apart from that, it has other roles like cell proliferation, differentiation, inflammation, angiogenesis, and immunity. The parathyroid gland plays a very, very vital role in the calcium phosphate metabolism. We have 4 parathyroid glands in our body. They are located in a superior and inferior pole of a thyroid gland. They are a highly vascular structure of size of 3 into 6 into 2 mm. Two types of cells are present in the parathyroid gland. Chief cells, they are the one which secretes the parathyroid hormone. Oxified cells are nothing but the degenerated chief cells. The parathyroid hormone is a linear polypeptide with a molecular weight of 9,500. It is made up of 85 amino acids. After its secretion, it lasts only for 10 minutes. After that, it gets disintegrated. Parathyroid hormone have effects on bone, kidney, and intestine. In bone, it mobilizes the calcium and phosphate from the bone to the bloodstream. The reabsorption of the calcium and the excretion of the phosphate will be happening in the kidney. It enhances the absorption of the calcium and phosphate from the GIT by increasing the synthesis of vitamin D. Parathyroid hormone and the vitamin D have an effect on bone. The parathyroid hormone has a two roles with regard with the maintaining of the calcium and phosphate level by mobilizing the two minerals from a bone. The first thing is it mineralizes the bone matrix by deposition of the calcium. Second thing is even it will mobilize the calcium and the phosphate from the bone to the bloodstream. Both the hormones will activate the osteoblast. The activated osteoblast will express the rank ligands that will go and interact with the pre-osteoclast. The pre-osteoclast will differentiate into an osteoclast. These are the cell mainly involved in a bone resorption. It has two roles: the removal of the minerals from the bone. Second thing is degradation of the bone matrix. The degradation is done by the proteolytic enzyme that comes out of the lysosomes. The acid that is secreted by the osteoclast are responsible for the removal of the bone, calcium, and the phosphate. When the serum calcium level falls, so the rapid release of calcium and phosphate will occur by the osteolysis of a bone by the osteocytes and the osteoblast. The slow release will be by the activation of the osteoclast. The other function of a vitamin D parathyroid hormone fibroblast growth factor 23 is reabsorption of calcium phosphate, excretion of a phosphate. Calcium and phosphate is reabsorbed with the help of vitamin D. The calcium is reabsorbed with the help of a parathyroid hormone. The phosphate is excreted under the influence of a parathyroid hormone and fibroblast growth factor number 23. The calcium absorption will be taking place in proximal renal tubule, thick ascending limb, distal convoluted tubules, and the collecting tubules. 90% of the calcium is reabsorbed in a proximal and thick ascending lobe. The remaining 10% is reabsorbed in distal convoluted tubules and collecting ducts. So the calcium will enter into the blood from the urine through the intercellular channels or by a transcellular mechanism. The phosphate absorption from the GIT is by either diffusion through the gastric cells or even it enter the blood through the intercellular spaces. The calcium absorption from intestine is mainly by the vitamin D. So under the influence of a parathyroid hormone. The parathyroid hormone is the one that helps in the conversion of a 25 hydroxycholecalciferol into a 125 dihydroxycholecalciferol. So which is very much essential for an absorption for a calcium and phosphate from an intestine. The absorption of calcium is by the mechanism by just fusion through the intercellular spaces with the help of a molecule called as a claudin, or through the facilitated diffusion with the help of molecule called as a colbidin or endocytosis. This type of transport is called as a vesicular transport. For the synthesis of a colbidin, vitamin D is required. So the vitamin D is the one that regulates the synthesis of a colbidin. So the calcium one bind to it. So together it is absorbed into the intestinal cells. The increase or decrease in the calcium level in the serum will determine the activity of the parathyroid hormone or parathyroid gland secretion. Decrease in the calcium increases the parathyroid hormone, so mobilize the calcium from the bone by resorption. The kidney, it increases the calcium reabsorption. When there is going to be a increase in the calcium level, the feedback mechanism will get activated. That will decrease the parathyroid hormone secretion. So how our body is able to sense there is an increase in a calcium or a decrease in calcium? This is mainly done by a receptor called as a calcium sensing receptor, which is distributed in the parathyroid gland and kidney. When there is an increase in the calcium level, this is done regulated by two factors, that is a parathyroid hormone secretion and the calcitonin, which comes from the C cells of a parafollicular cells of thyroid gland. This will have an opposite effect. It decreases the renal calcium reabsorption, decrease the bone absorption or bone resorption, then decreases the intestinal calcium absorption. When there is a fall in calcium level, then the parathyroid hormone secretion is increased. So it will increases the renal calcium reabsorption, then bone resorption and mobilize the calcium from bone to blood, enhances the intestinal calcium absorption by increasing the synthesis of 125 dihydroxycholecalciferol. Fibroblast growth factor is mainly involved in the phosphorus metabolism. It suppresses the phosphorus reabsorption by inhibiting the production of a sodium and potassium cotransporter, which is mainly essential for the reabsorption of phosphorus. Not only that, even it can influence the parathyroid gland as well as 125 dihydroxycholecalciferol. That is nothing but the active form of vitamin D. So next week we are all going to celebrate the great Indian festival Diwali. So happy Diwali, advance happy Diwali wishes to all my dear postgraduates. In making the fireworks also, the calcium and the phosphorus is used. The glowing nature of the crackers in a dark effect, that is mainly because of the presence of a phosphorus. When we burst the crackers, the orange color what we are getting, that is mainly because of adding a calcium chloride during its preparation. The interaction between the various organs like kidney, intestine and bone, it is regulated by various pathways. Any disharmony or dysregulation in the pathway can result in disorders like hypocalcemia, hypercalcemia, hyperphosphatemia and hypophosphatemia. The hypocalcemia is a condition where there is a decrease in a calcium level in serum. Hyper means elevated level. Hypophosphatemia means decrease in alkaline phosphatase level. When the vitamin D level decreases, then it is called as rickets in children and osteomalacia in adults. There is a special form of ricket called as vitamin D resistant ricket, that is otherwise called as a hypophosphatemia. This is mainly because of a chronic renal disorder. The other conditions what we can get is hyperparathyroidism, hyperparathyroidism and a pseudo hyperparathyroidism. Hypercalcemia can be because of a vitamin D deficiency or a decrease in the secretion of a parathyroid hormone. The vitamin D deficiency can be because of decrease in the synthesis or decrease in intake. So the synthesis of vitamin D is a multi-step process which involves a lot of enzymes. So the synthesis of the enzyme, if it is affected, definitely that have a effect on vitamin D synthesis. So there is a vitamin D deficiency condition caused by a genetic mutations of a hormone, so enzyme that is involved in its a synthesized pathway, like 1 alpha hydrolase, 25 hydrolase. Sometimes the receptor synthesis will be affected. So the genes that is responsible for synthesis of all these are mutated, that can result in a genetic mutated vitamin D. so which will ultimately result in a hypocalcemia. The common finding in all this genetic disorder or the genetic induced vitamin D deficiency is increase in the parathyroid hormone, decrease in the serum calcium and a phosphorus level. The baby suffering from rickets will show a features like a open frontal, stunted growth, odd-shaped skull, a spine deformities, wide elbow, wrist, knee and ankle joints, ball legs, pelvic deformities. So even they'll have a pigeon chest. So this is a picture of a child with a ricket. You can see the deformities in the upper and lower extremities. So because of a poor mineralization, there'll be coughing and expansion of metaphysis in a patient suffering from rickets. The anterior end of the ridges can go for a bulbous expansion that is called as a rickety rosary. So these are the common findings that is seen in the children suffering from a rickets. The vitamin D deficiency in adults can result in a condition called as osteomalacia. This is highly prevalent in Indian, Japanese, and Chinese, especially in the females who are in a postmenopause period. The main cause for getting osteomalacia is lack of sun exposure and decrease in intake of vitamin D. So they'll have a symptoms like excess sweating, weight loss, mood swings, joint pains, dry skins, and then frequent fractures. There'll be a reduction in their height. They have a difficulties in walking. The weakness of the limbs are reported. Even there'll be a yearly tooth loss. There is another form of ricket called as renal rickets that is commonly seen in end stage disorders, mainly seen in people those are suffering from a chronic renal disorder. This is an X-ray of a patient suffering from a ricket showing a widening of radiolucent growth plates due to a poor mineralization that is seen as a playing and popping of a metaphysis. Individuals with an osteomalacia will show a looser zone in their extremity bone, sometimes even in other areas. So here you can see the hand showing a looser zone with the sclerotic margins in the radia and ulnus. So these bones are very, very vulnerable for a fracture because of the looser zone. The osteomalacia is mainly because of a nutritional deficiency and indoor activities. The another reason for getting a hypocalcemia is a hypoparathyroidism. So in the hypoparathyroidism, there is a marked decrease in the calcium concentration and increase in the serum phosphate level. So the parathyroid hormone level will be very low or even it is undetectable. So even the vitamin D level will be markedly decreased. The reason is the vitamin D synthesis is very much depend on a parathyroid hormone. So since the hormone level is reduced, even the vitamin D level is also decreased. So the patients with the hypothyroidism will have a decreased renal filtration load of a calcium, and they have a low nephrogenesis cyclic AMP excretion. The renal tubular reabsorption of a phosphate is elevated in them. The sole cause or the main cause for getting a hypoparathyroidism in adult is thyroidectomy. Following a thyroidectomy, so the parathyroid gland also will be removed. So this is the treatment option for some of the tumors of the thyroid gland. So since it's a tiny gland, so removing that gland alone is not possible. So along with the thyroid gland, even the parathyroid gland will be removed. So that will result in a severe hypoparathyroidism. Even genetic factors contribute for a hypoparathyroidism in children's. So when there is going to be a mutation of a calcium sensing receptor, so that can result in a hypoparathyroidism. So the plasma parathyroid hormone level will be markedly decreased. The calcium level also will be decreased, but the phosphate level will be, or the phosphorus level will be much elevated in the individuals with with the hyperparathyroidism. There are subtypes based on which type of gene that is involved. There is a condition called as a pseudo hypoparathyroidism. Here, there is an excess secretion of a parathyroid hormone, but there is a defect in its action, so which results in a decrease in a serum calcium level and a serum phosphate level. This happens when there is going to be a defect in their receptor or a mutation of the gene that is coding for the receptor or the other cell signaling molecules. Some of the symptoms of a hypercalcemia or a numbness over the oral cavity, paresthesia of the distal extremities, muscle cramps, carpopedal spasm, tetany, then there'll be positive chopstick sign, laryngospasm, bronchospasm. Some people even they'll experience seizure when there is a fall in calcium level. The other symptoms are a fatigue, irritability. So even sometimes it will become a life-threatening condition because of arrhythmias. People with a chronic hyperparathyroidism will show a calcification in the basal ganglion or intracranial calcification that will be well evident in X-rays and CT scans. So some people will experience neurological symptoms because of intracranial calcification. So the other symptoms includes subcapsular cataracts, abnormal dilation, alopecia, post-brittle hair. If a hypercalcemia state occurs in the young stage or during the childhood period, even the tooth will show a lot of hypercalcified areas. So some of the dental abnormalities that is observed in a hyperparathyroidism is a dental enamel hyperplasia, delayed eruption, hyperdontia, malformed tooth, polycalcified dentin, widened pulp chambers, even pulp calcification will be evident and pulp chamber calcification will be evident in some individuals, malformed roots, dental malocclusion, ankylosis, they are very vulnerable for caries. Some people are very prone for candidal infections. The paresthesia of tongue and lip is also reported. So let me move on to a next condition called as a hypercalcemia. So this is an increase in the serum calcium level, just opposite to the hypercalcemia. 1 in 500 among the general population is affected by a hypercalcemia. The three reasons for getting a hypercalcemia is malignancy, hyperparathyroidism, and increased in the synthesis of vitamin D. Parathyroid hormone related protein is produced by many body tissues which have 140 amino acid residues. The interesting thing about this protein is it has a similarity with the parathyroid hormone. So it will have the similar effect like a parathyroid hormone, but only the 8 amino acids are in the same position like a parathyroid hormone in this protein. The hypercalcemia, what we get because of a malignancy, can attribute by two factors, or it is by two factors. One is local osteolytic hypercalcemia. So because of local invasion or a bone metastasis, there will be hypercalcemia in 20% of an individual, or elevated level of parathyroid hormone related protein released by the tumor cells of a breast, kidney, ovary, and skin. The calcium and the phosphate metabolism, even it is influenced by other hormones like insulin, estrogen, thyroid, growth hormones. Insulin, for example, increase the density of the bone. Estrogen is very much responsible for the synthesis of the bone matrix in its decreased level. So it results in a condition called as an osteoporosis. Hyperparathyroidism, even caused by a genetic mutation of the calcium sensing receptor, a genetic mutation of a proteins that is called as menin, and then red CDK1B. So mutation of this multiple genes can result in different hyperparathyroid conditions like familial isolated hyperparathyroidism, hypothyroidism, jaw tumor syndrome, neonatal severe hyperparathyroidism, men 1, men 4. So these are the variety of diseases or the disorders that is occurring because of the mutation of the genes that is involved in the parathyroid hormone synthesis, parathyroid hormone sensitization, and the parathyroid hormone effects. The interesting thing here is there is an excess production of a parathyroid hormone, so which results in a decrease in a serum calcium level and increase in a serum calcium level and decrease in a phosphate level. There are two types of hyperparathyroidism, primary hyperparathyroidism and a secondary hyperparathyroidism. The cause for a primary hyperparathyroidism is a tumor of any one of the parathyroid gland. It is frequently seen in men when compared with men and children. The symptoms that is experienced by the hyperparathyroid thyroidism patient is fragile bone osteoporosis. The bone will be very, very vulnerable for a fracture, kidney stones, and then metastatic calcification. Calcification will be occurring in the soft tissues because of an increase in a serum calcium level, excess urination, abdominal pain, easy tiring, weakness, bone and joint pains. Some psychiatric symptom also they'll be experiencing like orientation, disorientation, delirium, confusion, hallucination, forgetfulness, alteration in hunger. The secondary hyperparathyroidism is mainly because of a parathyroid gland hyperplasia. This occurs when there is a deficiency of vitamin D and a chronic renal disease. So the vitamin D is essential for the absorption of calcium and phosphate for a GIT, and the renal system is essential for the reabsorption of the calcium and the excretion of the phosphorus. When there is going to be a vitamin D deficiency and the chronic renal disease, the calcium phosphorus level will be affected. There will be alteration in this level. This is sensed by the parathyroid gland. So to compensate the decrease in the calcium level, the gland will go for a hyperplasia. So there will be excess secretion of a parathyroid hormone, so which will have a adverse effect on a bone by increasing the osteoclastic activity. So which will result in decalcification. There will be punched out cystic areas of bone which will be filled with an osteoclast that is called as a Brown's tumor. These people are very, very vulnerable for a fracture because of weakening of a bone. So the punched out radiolucencies, large radiolucency seen in the extremities as well as in the facial bones is called as the osteitis fibrosa cystica. The dental abnormalities that is observed in hyperparathyroidism are obliteration of pulp chamber, alteration in the dental eruption. So there will be a premature eruption of the tooth, loosening and drifting of the tooth. Some people have malocclusion, spacing. So there will be loss of lamina dura. So it will be generalized. Periodontal ligament widening will be there. So the tooth will be very, very sensitive for a percussion because of the presence or development of a large cystic areas. Vulnerable for a fracture because of weakening of a bone. So the punched out radiolucencies, large radiolucency seen in the extremities as well as in the facial bones is called as the osteoarthritis fibrosa, cystica. The dental abnormalities that is observed in hyperparathyroidism are obliteration of pulp chamber, alteration in the dental eruption, so there will be a premature eruption of the tooth, loosening and drifting of the tooth. Some people have malocclusion, spacing, so there will be a loss of lamina dura, so it will be generalized. Periodontal ligament widening will be there, so the tooth will be very, very sensitive for a percussion because of the presence or development of a large cystic areas. Floating tooth appearance will be there. So these large cystic areas will show a collection of numerous germ cells under the microscope. That is called as a Brown's tumor. So the Brown's tumor is the one that is developed in a secondary hyperparathyroidism. The name why it got is so it appears brown in color under a gross specimen. So there will be a soft tissue calcifications in a hyperparathyroidism. So development of a salivary calculi in condition in patients with a hypercalcemia, which occurs because of a hyperparathyroidism. Even some individuals, mandibular tori is report. Even bone pain, the jaw bone pain is also is very common in patients with the hyperparathyroidism. Hypophosphatasia is a condition because of a decrease in the enzyme alkaline phosphatase. So alkaline phosphatase is an enzyme which plays a major role in bone mineralization. So the deficiency of this one can result in a condition called as a hypophosphatasia. So the clinical presentation is similar like a vitamin D resistant ricket. In addition to that, patients will have a premature exfoliation of the deciduous tooth and they are very, very vulnerable for infection. Growth retardation is a common finding in patients with the hyperphosphatemia or a hyperphosphatasia. So this is a condition commonly reported in this is a very, very rare condition. So the symptoms will be similar like a vitamin D. So in addition to that, they have a disorders of a kidney, GIT and lungs. So the metaphysis of the long bones under the imaging shows spotty, streaky or irregular ossifications. Dental X-ray usually shows a hypercalcified roots and a large pulp chambers. The problem with the hypophosphatasia or a decrease in alkaline phosphatase is because of the mutation for the gene that is coding for this enzyme. So the abnormalities what we can observe in the serum is the parathyroid hormone level will be decreased, the serum phosphate level will be elevated, the serum calcium level will be elevated, the vitamin D synthesis also is very much downregulated in this condition. So diagnosing of all the disorder associated with the calcium and phosphate metabolism is by a proper clinical examination and subjecting them for a serum and urine analysis and imaging the skeletal system. So by correlating all these things, a final diagnosis will be arrived and the treatment for all this disorder is to remove the etiology. That is the primary thing. The secondary thing is we can give vitamin supplements to strengthen the bones and to regulate the levels of the calcium and the phosphate in the serum. So in the deficiency sites, even the parathyroid hormone is given for the patients. Sometimes even the phosphate is given in some conditions where the phosphate level is very much decreased. For example, hypophosphatemia. So that is a condition where there will be a decrease in a serum phosphate level. So with this, I finished my assignment. Okay, thank you, my dear postgraduate, for your kind listening.
Thank you, ma'am. Your delivery was so engaging that we lost the track of time, ma'am. Providing us an expert knowledge on this topic, we thank you, ma'am. Queries will be answered before moving forward to next questions. We have few questions, ma'am. Can I go? Am I audible, ma'am?
Madam, kindly unmute your. Oh, yeah.
Yeah, I have
Am I audible?
muted my mic. Yeah, you are audible.
Ma'am, we have few questions. Can I ask, ma'am?
Yeah.
One of the participant is asking correlation between the fibrous dysplasia and serum PTH and alkaline phosphatase.
Actually, fibrous dysplasia is a different entity. It is a fibrous condition where the bone will be replaced by a fibrous tissue. Later on, ossifications will come in that. Okay, so there also the bone is going for a remodeling. But this is not because of an elevation in a parathormone. So that is because of a specific mutations like G protein mutation. So the serum elevated level is elevated when it is of a polyostotic type. And then alkaline phosphatase level is also elevated in some cases. The reason is that is the enzyme that is involved in the bone formation. So patients with the fibrous dysplasia, they'll be actively involved in the bone synthesis. So the serum calcium level as well as the alkaline phosphatase level will be elevated in them, but the parathyroid hormone level will remain the same.
Thank you, ma'am. We have another question. What are the major oral manifestations we can see in vitamin D deficiency patients?
Vitamin D deficiency patients, I told you it is needed for the mineralization. So when the if the deficiency occurs during the tooth development stage, we can make out lot of hypocalcified areas. Malformed tooth will be there. So a lot of irregular surfaces we can we can see on the surface. I mean on the enamel surface.
Thank you, ma'am.
Thank you so much, ma'am. Nice seeing you after a very long time.
Nice seeing you, Shweta. I should thank the Vishnu team for giving me this opportunity.
Thank you so much.
So everything is possible. Everything is possible because of you, Shweta. So a special thanks for you.
Thank you so much, ma'am. My best wishes to the entire team of our oral pathology Annamalai University.
Thank you. Happy Diwali wishes for the Vishnu team and all the postgraduate who are in link now.
Yes, ma'am. Thank you. Same to you, madam.
Thank you.
Ma'am, we have a token of appreciation from VISHNU ADHYAAN, ma'am. Please accept the certificate. We'll display it.
Yeah, sure.
Next lecture is going to be given by Dr. Vinita Gupta ma'am on the topic of bone pathology. Vinita Gupta ma'am currently working as a professor and the head of the Department of Periodontics in Government Dental College, Raipur, Chhattisgarh. She has a vast experience in field of periodontics from 19 years. She is selected by Indian Council of Medical Research for workshop on research methodology for dental professionals among 20 dental professionals selected all over India. She is the one of the finest subject expert in various dental institutes all over the India. She has more than 50 national and international papers to her credit. She has been a DCA inspector for various government dental colleges throughout the India for UG and PG recognitions. I hand over the platform to you, ma'am. Thank you.
Morning, everyone. Bone biology is the topic for today. I'm Dr. Vinita Gupta, heading the Department of Periodontics in Government Dental College, Raipur. Special thanks to Dr. Gautami and her energetic team for giving me the opportunity to present the seminar on on the platform of VISHNU ADHYAAN 2022. A special thanks to Dr. Divya for her continuous support. This is my team from Government Dental College. Bone has been an integral part of all dental specialties. For example, in the field of perio, we learn more about bone biology. We learn about the bone defects, their regeneration, and in implants, we are more concerned about the osseointegration and for the implant stability and its long-term prognosis. Being a prosthodontist, they'll be more concerned about the resorption pattern in the edentulous patients, about the patterns of bone growth, bone remodeling as such. And orthodontists will be more interested in growth of the bone, the theories of the bone, so that they can take the advantage for moving the tooth and aligning the tooth likewise. So starting the presentation, I'll be covering the topic sequentially. The table of contents will be like bone, its classification, composition, development, and bone in health and disease. Bone and its function, it's a specialized form of connective tissue with calcified intercellular substance. It provides support, it acts as a framework, it provides protection. That is, the non-mobile joints of our body, like example, skull and the vertebrae, they surround the soft tissue and they provide protection. Movement, some of our joints of the skeleton are mobile and they act as levers to move the body. So it's for storage and the blood cell formation. We are all well versed with it. Coming to classification, developmentally, it can be classified as endochondral and intramembranous bone, histologically as compact and spongy bone, and by shape, it can be classified as long, short, flat, and irregular. Composition, basically, it has inorganic component of 67%, and mainly it is hydroxyapatite. Organic is 33%, non-collagenous protein 5%, and the collagen, basically, it is of type 1 has type 1 collagen, and minimal amount of type 3 and type 5. Development of bone, the interesting fact is that maxilla and mandible both develop intramembranously, and it is the product of the first arch epithelium during the second week of the intrauterine life. And the point to be noted is the size of the alveolus is dependent on the size of the growing tooth germ. After the initial development of growth of alveolar bone, it is governed by the ectomesenchymal tissue around the early tooth germ. So the alveolar bone is ectomesenchymal in origin. The tooth moves into place, developing its own alveolar bone from its own follicle. This is the developmental chart. The tooth germ that further differentiates into enamel organ, dental papilla, and the dental follicle, which further differentiates into cementoblast, osteoblast, and fibroblast. And osteoblast will help in the alveolar bone formation. Classification of alveolar bone depending on the functional epithelium, the mineralization, and the ideal bone structure was described by Linko. He classified it as the ideal bone consisting of the evenly spaced trabecular with small cancellated spaces. Lancome and Zarp, mostly we follow this. The quality 1 bone, that is the homogeneous compact bone. Quality 2 bone is the thick layer of the compact bone surrounding a core of dense trabeculae bone. Then the cortical bone gets thinned, and this is in the quality 3 bone, and surrounding a dense trabeculae bone of favorable strength. And here in the quality 4, the cortical bone is even thinner, and the core of the trabeculae inside it is of low density. So mainly in the mandibular anterior areas, we find the quality 1 bone. Here the vascular supply is compromised as when we are concerned of osseointegration, we are placing the implants, and the quality 2 and quality 3 bones are more favorable for as concerned to the vascular supply. Mesh, the density classification, everyone is well about it. These are the types of the bones that we can find in the maxilla and the mandible. Type 1 basically in the symphysis area. Here the type 2 on the and here type 3. Alveolar bone process, or the alveolar bone, it is a part of the maxilla and the mandible and has distinct structural, functional, and developmental characteristics. It consists of the cortical bone, the cancellous bone, and the alveolar bone proper. Now, this is the outer alveolar plate, the inner alveolar plate, interdental septum, and the interradicular septum. This is the alveolar bone proper that surrounds the root of the tooth or the socket of the tooth. These are the cortical bone. This is the cortical plate or the cortical bone, and in between is the cancellous bone. Alveolar bone proper, otherwise if the radiological term, we can also call it as lamina dura. It is a thin lamina of bone that surrounds the root of the tooth and provides attachment to the principal fibers of the PDL. The cribriform plate, it's perforated and carries branches of the interalveolar nerves. And here we see the bundle bone, that is the histological term. Inner surface is known as the bundle bone because abundance of PDL sharper fibers are inserted into the bone. The cancellous bone, it surrounds the alveolar bone proper and gives supports to the socket. In maxilla, we see the type 2 pattern, that is the irregularly arranged numerous delicate interdental and interradicular trabeculae. And here in the mandible, we see the type 1 bone pattern, that is a regular arrangement is seen in a horizontal manner, and it's like a ladder-like arrangement. Talking about the alveolar crest, it's a junction between two teeth, and here the alveolar bone proper meets the interdental septum, as well as the cortical plates also meet here. And it is thin, knife-like, edged, and scalloped, especially in the anteriors, but it is rounded and beaded towards as we go to the posterior teeth. In health, the alveolar crest extends to approximately 1.5 to 2 mm from the CEJ. And the thing that is to be noted is the positive architecture that it has. It is the height of the interdental bone is more at the coronal level as compared to the labial or the lingual bone. Interdental septum, it is the bony portion that separates the adjacent alveoli. If the roots are too close together, an irregular window can appear in the bone between the adjacent roots. The interradicular septum, the bone between the roots of the multi-rooted teeth, and they contain the nutrient canals. The applied part is interesting, and the height and the thickness of the bony plates can be affected by the alignment of the teeth, angulation of the roots to the bone, and the occlusal forces. The thinner labial plate is comprising of the bundle bone that is vulnerable to both resorption and the iatrogenic damage. And frequently it is lost by traumatic extraction, restorative procedures. The cortical plates, as we had discussed earlier, it is thicker in the lingual and the palatal surface as compared to the buccal surfaces, and thinner in the maxilla than in the mandible. So the structural anatomy and the thickness of the cortical bone will depend to some extent on the position of the teeth in the arch. The cortical bone is covered on the outer surface by the periosteum. Periosteum is a dense, irregular fibrous, is of dense, irregular, and has it's of fibrous layer. It also has two layers. That is, the outer layer is the fibrous layer, and the inner layer is more has more regenerative potential as compared to the endosteum, that is the inner layer. Seeing the histology of the bone, these are the primary anatomical function units of the bone, the osteon or the Haversian system, and that contains the vessels, the Haversian canals. Here we can see the Workman canal and the laminae and the lacunae and the canalicula. The Workman canals adjacent the Haversian they connect the Haversian systems, and they are perpendicular to the central canal and contains the vascular network. The outer perimeter is covered by the circumferential lamellae, and it encloses the entire adult bone. Here we see the concentric lamellae that makes up the bulk of the compact bone and is the basic metabolic unit of the bone or the osteum. The interstitial lamellae, here we see the interstitial lamellae in between, that is interspread between the adjacent concentric lamellae and fill the spaces in between. Here there's a clear picture of the Workman canals connecting the Haversian and the concentric lamellae together. And these are carried to the Workman canals are carried along with the Sharped period to the period, giving the vascular supply. The fenestration and dehiscence, these are the defects, roughly 20% of the anatomical defects of the alveolar process. These are the fenestration or the windows that here the marginal bone is intact. And in dehiscence, the marginal bone is lost, and the defect is through and through. We can see the bone loss. These defects can complicate the therapy when root coverage procedure or implant is attempted. And they can also influence the pattern of bone loss in periodontal disease. So, seeing the histology of the compact bone and the spongy bone, it is comparatively hard and compact, and it is spongy in nature. Lamellar pattern here in compact bone is regular, and in spongy we don't have a regular pattern. Bone marrow is yellow in the compact bone, especially in the adults, and a spongy bone has the red bone marrow, so it produces RBCs all the time. And in adults, the red bone marrow is found in the alveolar bone. We find it usually in the maxillary tuberosity, maxilla, and the maxillary and the mandibular molar and the premolar areas. Lower molar, so while taking the graft, we should keep this in mind. Lower marrow content means that the intraoral sites are not great source of osteoprogenitor cells, which may be the reasons for suboptimal performance of the graft sometimes. Osteogenesis, it is the process of bone formation. The bone formation can be of two types: intramembranous ossification and endochondral ossification. In the intramembranous ossification, it occurs by direct differentiation of mesenchymal cells into the osteoblast, which in turn lays down the bone matrix. But in endochondral ossification, it occurs by the invasion of the osteoblast into the cartilaginous template. These are the cells of the bone, and this is osteoblast. It especially, its main function is to form the matrix, and cuboidal in shape with a single nuclei, and acts on the bone structure. The cell lineage is basically, it is a mesenchymal in origin, and produces collagen type 1 alkaline phosphatase. Here, these are the osteoclasts. Their basic function is to resorb the bone or the bone matrix, large multinucleated cells, and acts on the bone. And this is the active form because it has the ruffled border. It is hemopoietic in origin and secretes the key products are MMPs, acid, and cathepsin K. These are the osteocytes. They are the cells embedded, osteoblasts, that are embedded in the bone matrix. It has a single nuclei and has numerous projections. You can call it as a teenage stage of osteoblast, and this is the grandfather of the old age stage of the osteoblast, that is the osteocyte. And it is also mesenchymal in origin. To keep up the functional demands, the alveolar bone proper has to undergo constant remodeling, and this is achieved by continuous cellular activity in the alveolar bone. So, talking about remodeling, remodeling helps the bone to respond to mechanical stresses and repair wounds and maintain the calcium and the phosphate homeostasis. And parathyroid hormones play a regulator for important part in regulating the bone remodeling. Seeing the here we see the calcium homeostasis. This is the parathyroid glands and the thyroid glands, which play an important role. Here, when we see a decrease in the calcium level, that leads to the secretion of the parathyroid hormones, and which in turn stimulates the osteoblast. The osteoblast in turn releases interleukin-1 and interleukin-6, which stimulates the migration of monocytes to the sites of the resorption. And the osteoblast also secrete the rankle, and the rank and the rankle interaction takes place, and then that signals for bone resorption. So here we see an increase in calcium resorption. Formation of calcitrol is also increased. Calcium increase, ions increase, absorption is increased. Bone resorption and the osteoclastic activity is increased, and there's a decrease in the calcium excretion. When the calcium levels are normal again, so there's a signal to the thyroid gland where the calcitonin is released, secreted, and it acts upon the kidney, small intestine, and various other organs. And here we see calcium resorption, phosphorus excretion is increased, calcium absorption is decreased, bone resorption is decreased, and calcium excretion is increased. We'll take up with the diagram here. This is the hemopoietic stem cells. This is how the differentiation takes place. When there's a decrease in the calcium level, the osteoblasts release the interleukin-1 and interleukin-6 that stimulates the monocytes to reach the site of the resorption. And the osteoblasts secrete the nuclear factor kappa B ligand, that is rankle, and rankle binds to the receptor activator rank in the pre-osteoblasts. It causes the breakdown of the bone matrix, and the matrix releases various osteogenic substances such as the bone morphogenic protein, resulting in the form of multinucleated osteoclast. Thus, in turn, stimulates the differentiation of the osteoclast, and here the osteoclasts are activated with the ruffled border, creating an acidic environment for bone resorption. When the calcium is retained, the osteoblasts are activated, and the rankle now binds to the osteoprotegrin, and here the osteoblastic activity, that is bone formation, starts. So there can be three fate of the osteoblast, that is, it can go for apoptosis, lining cell, can become a bone lining cell, or it can become an osteocyte, or that is, it gets embedded in the lamellate cell. This interdependency is called as the coupling mechanism. The modeling has different phases, that is the quiescent phase, activation phase, resorption phase, reversal phase, and the mineralization phase, followed by the quiescent phase. The phases like activation phase, here the signals are being given to the osteoblast is giving the rankle, and here it is activating the osteoblast, and here osteoblast, and the ruffle border is seen. The bone remodeling is taking place. Resorption phase is continued. Then is the reversal phase. Osteoblasts are activated. Osteoid, that is the bone formation, is done. Mineralization by the deposition of the hydroxyapatite crystals, and then again the quiescence phase. Stages of healing, there are 4 stages, and there are multiple stages of healing. Stages of hematoma, granulation tissue, callus formation, remodeling, and modeling. This has been taken up by Dr. Vandana ma'am, and talk about in brief. Healing and regeneration, it can be assessed and evaluated for the comparison of different modes of therapy. The primary methods that we can use for evaluation are the histological analysis, that is the gold standard, and we can go for clinical examination, radiographic analysis, biochemical analysis. Others are the fluorescent labeling for cementum and wound healing indices. Theories of growth, the bone remodeling theory, genetic theory, sutural theory, cartilages, and various other theories that we can read up for growth of bone. We usually follow the Wolff's law when we apply in orthodontics, that is the tension and the compression cycles that are created, and that in turn creates a small electric potential that stimulates bone deposition and increased density at the points of stress. Here we see the tension area where osteoblasts take the position, and in the pressure area where the osteoclasts take the position and bone resorption, and this initiating the movement of the growth. Clinical implication, so the mechanical load is necessary for bone remodeling. Absence of mechanical stress results in decrease in the bone density, and bone responds to pressure by resorption and to tension by bone apposition. If heavy forces are placed, bone generally responds by increasing the number of the trabeculae, and these trabeculae are arranged perpendicularly to the vector of the forces. This can be applied when in cases of periodontal flap surgery. Sometimes you may have seen the bulbous bone pattern. So that is the defense mechanism of the body itself. Regulatory factors in bone remodeling. I have seen short questions being asked on this topic. The systemic factors that are responsible for decreased bone resorption are calcitonin and estrogen hormone. Local factors, growth factors, various growth factors can be responsible for increased bone resorption, parathyroid hormone, glucocorticoids, and vitamin D in high doses. And cytokines also can be responsible, like example, interleukin 1, 6, interleukin 11, and PGE2. For increased bone formation, growth hormones, vitamin D, insulin, and low dose of parathyroid hormone can increase the bone formation. And glucocorticoids can be clubbed with the decreased bone formation. Markers of bone turnover in the serum markers are alkaline phosphatase, osteocalcin, and collagen 1. The markers of bone resorption in urine are urinary calcium, hydroxyproline, collagen cross-link fragments, and the urine protein, pyridinoline. Seeing the osseous topography, in health, normally the roots with the intervening vertical depression that tapers towards the margin. And in the facial labials version, the margins of the labial bone is thin to a knife edge and present an accentuated arc in the direction of the apex. On the lingual version, the margins of the labial bone is blunt and rounded and horizontal rather than arcuate. The arc, here the horizontal pattern is seen. Okay. Blood supply and the nerve supply, it is derived from the inferior and the superior alveolar arteries, and the venous drainage accompanies the arterial supply. And nerve supply is basically from the anterior, middle, and the posterior superior alveolar nerves for maxilla. and branches from the inferior alveolar for the mandible. It is like when we are harvesting the autogenous graft from the mandible or from the maxilla, we should be knowing from the blood supply properly so that we don't land up with hemorrhages or paresthesia that may be a complication later on and is difficult to deal with. Age changes that we see, they have the periodontal surface of the bone become more irregular, and the bone alveolar bone appears more jagged or uneven, and a less irregular insertion of collagen fiber is seen. And osteoporosis ensues with aging, with decreased vascularity, and the red bone marrow that we had earlier studies is now filtered with yellow bone marrow that has fatty infiltration, and there's a reduced metabolic rate and healing capacity. The point to be noted is the bone graft preparation from the older donors, they have lesser osteogenic potential than the graft as compared to the younger donors. And the other factors to be taken care is the calcium and vitamin deficiency in the older adults. In the menopausal female also we can see bone loss. Age, genetics, diet also play an important role in the decreased bone mass and the bone quality. And it may lead to multiple fractures, and trauma can also be a cause to that. In edentulous jaws, attrition and the loss of alveolar may cause increased wrinkling around the mouth and a decrease in the vertical dimension. So mostly in the adults, they usually go for frequent relining of the dentures. Alveolar bone in disease. The condition involving loss of the alveolar bone are the extension of the gingival inflammation, trauma from occlusion, and systemic factors. And the local factors we can talk is periodontitis, periodontal abscess, food impaction, overhanging restorations, adjacent tooth extraction, or ill-fitting prosthesis. The bone destruction caused by extension of gingival inflammation. The gingival inflammation also can be a factor. The inflammation can be directly from the interproximal surface. The gingiva inflammation can directly go to the bone or to the supporting structures, or from the bone to the supporting structures. Here we can see the inflammation has traveled to the supporting pedial fibers or to the periosteum, and from the periosteum it is going towards the bone. The mechanism of bone destruction, normally bone is in a hemostasis state where the osteoblast and the osteoclast coupling mechanism is not disturbed, and this coupling is disturbed only when there is an excessive osteoclastic activity, maybe due to result of direct effect of plaque products on the osteoclast, or effect of plaque products on the gingival fibroblast and the immune cells, which in turn releases pro-inflammatory mediators. And these pro-inflammatory mediators, as I've already discussed, it can be interleukin-1, 6, PGE2, or lipopolysaccharide, which influence the gingival fibroblast, osteoblast, and T cells to release rankle, which binds to rank on the osteoclast and thus activating the osteoclastogenesis. The radius of action, as suggested by Grant and Cho, suggests that bacterial plaque can induce bone loss within the range of 1.5 to 2.5 mm, and the rate, as according to that the classical study on Sri Lankan labours, it is 0.2 to 0.3 mm in a year. The bone loss, the point here to be noted is the bone loss caused by trauma from occlusion. It can be modified by the presence or the absence of the inflammation. And in presence of inflammation, the bone loss tends to be faster, and the nature also changes to vertical bone loss. The different graft materials that we use for regeneration and in the defect areas are autograft, allograft, xenograft, and the alloplast. And here we have to take into consideration the bone regeneration potentials of different grafts as different, and while we are using it, we have to remember the resorption time, and we have to keep in mind which graph we are using in which site and for what purpose. So different GART material, they can go for regeneration, with different mechanism, like they can go for osteogenesis, osteoconduction, or osteoinduction, depending on the nature of the graft material. Bone destruction pattern in periodontal disease, most commonly we see a horizontal bone loss. So what is a horizontal bone loss? It's like the bone is reduced in height, but margin remain almost perpendicular to the tooth surface, not necessarily equal degree around the same tooth. Other bone defects are, these are the periodontal osseous defects. Here we can see most of the students have confusion here. This is the 3-wall defect, 1, 2, and the 3 walls that are seen here. The number of walls that are present, accordingly, we classify the defect. Here are there 2 walls seen, it is a 2-wall defect, and here only 1 wall is there, so it's a 1-wall defect. And we usually study about the supra-bony defects, but supra-bony defect is not an appropriate, or we can say it is a misleading term, because no defect exists above the bone level. These are the various other types of defects. The vertical defects commonly seen, and we have earlier discussed, if TFO is there and it is combined with periodontal inflammation, the horizontal bone pattern is changed to a vertical defect. It can also be seen with aggressive periodontitis, port impaction, and trauma from occlusion. This is a hemiseptum. There are vertical defects with only 1 wall of the alveolar bone present in the interdental area. And usually it has a poor prognosis. This is the osseous crater that are bounded with the facial and the lingual walls. And these we can appreciate the reverse architecture, that is the interdental bone is on a apical level, not the coronal level, as compared to the facial and the lingual bones. This is the buttressing bone. It may be found on the facial aspect and usually occurs as an adaptive remodeling to the occlusal forces. Bone destruction may be due to systemic diseases. I am covering the important diseases, the common one. The vitamin D deficiency due to diabetes, hyperparathyroidism, hematological disorders, Paget's, fibrous dysplasia, and osteomyelitis, giant cell granuloma, aneurysmal bone cyst, and others. In vitamin D deficiency, generally radiologically we see generalized partial to complete loss of lamina dura and reduced density of the supporting bone, loss of the trabeculae, and increased radiolucency of the trabecular interstices, and increased prominence of the remaining trabeculae. In diabetes, Taylor et al. Had suggested that poorer glycemic control leads to both an increased risk for alveolar bone loss and more severe progression. Hyperparathyroidism, here we see oral changes that include malocclusion and tooth mobility, radiographic evidence of alveolar osteoporosis with closely meshed trabeculae, widening of the lamina dura, and radiolucent cyst-like spaces can be seen. The other diseases in which it may occur are Paget's, fibrous dysplasia, and osteomalacia. In Paget's disease, the normal trabeculae pattern is replaced by haze, and diffuse meshwork of closely knit fine trabecular markings can be seen with lamina dura absent. Or scattered radiolucent areas may contain irregularly shaped radioopaque zones. Hematological disorders, here for example, in leukemia, the presence of infiltrate in bone marrow spaces and the periodontal ligament results in osteoporosis of the alveolar bone with destruction of the supporting bone and disappearance of the periodontal fibers. Excuse me. In sickle cell anemia, many cases are there in Chhattisgarh, so we are much interested in sickle cell anemia. We see a peculiar step ladder alignment of the trabecule of interdental septum. These are the cases of osteomyelitis. A case of chronic osteomyelitis of the jaw. So we have to have a sound knowledge of health and disease. So we can differentiate between what is develop a accurate treatment. Bone and soft tissue healing in dental implantology. The knowledge of bone and soft tissue healing can be applied to enhance the success of dental implants. And here osseointegration plays an important role. Because it depends on the vascular supply at surgical site and the primary stability of the endogenous implants. Healing in dental implantology is all the same. Like it starts with the inflammation, and there's the resolution and bone formation, followed by remodeling and maturation. Osteointegration, it depends on the factors that can play a role in osteointegration are biomaterial, implant design, biomechanical factors, its surface characteristic, the surgical technique, and last but not the least, the health and the quality of the bone. And factors that influence the implant stability, first and foremost, is the bone quality and the bone quantity. Secondly are the other factors, and for secondary stability also, the bone remodeling and modeling are important factors to be considered. The drugs that show an impact on bone health are glucocorticoids, aromatase inhibitors, androgen deprivation therapy. PRP has taken a lead role for bone in therapies for bone regeneration, for muscles also, for bone and cartilage regeneration. We are nowadays using PRP and different forms of PRP. Lasers also have taken nowadays the advanced technique for treating bone cancer, implant related infection, for example, in periimplantitis and perimucositis. Also, they have been opted for arthritis and for bone regeneration. To conclude, alveolar bone forms an architectural base for the healthy periodontium. So we should not circumvent the biological and the physiological events taking place during surgery and healing that may lead to the failure of the treatment. Integration, accuracy, biological skills are key elements to success of any treatment. So to understand Mother Nature and act accordingly, the probability of success will surely be higher. Thank you. These are my references.
Good afternoon, Dr. Vinita.
Good afternoon, Dr. Gautami. Nice to see you.
Same here. Very nice presentation on bone biology. I was listening to the presentation. Yeah, and I think you have a couple of questions.
Okay. Ma'am, can I ask you, ma'am?
Yeah.
Yeah, one of the delegate is asking, please can you tell me about bone morphogenic proteins, ma'am?
Bone morphogenic proteins, it's released by when the resorption takes place. There's a degradation of the organic and the inorganic materials. So they release the bone morphogenic proteins, then in turn signals the osteoclast again, and then the coupling mechanism starts. The calcium ions are sufficed, and the osteoblastic initiation takes place, and bone formation can take place further. So this is an osteoclast and osteoblastic coupling mechanism, and the BMP signals the process. And detail of the bone morphogenic proteins can be dealt separately. It's a different topic altogether. Here, the role is basically of signaling.
Ma'am, there is
other point. Yes.
Can you please elaborate on the dental considerations in patients with osteoporosis?
When we are dealing with osteoporotic patients, we should first take a complete detailed history of the patient. The bone density mapping should be done. And then accordingly, we should plan our treatment. And it's not that it is a no to regenerative treatment, but seeing the status of the levels of the calcium and phosphorus ions, we can plan accordingly. And it's an indicated treatment we can take. We can opt for vitamin D substitutions, and further calcium also can be monitored on a monthly or a routine basis.
Thank you, ma'am. I appreciate your dedication, ma'am. Despite of being not well, you have delivered an excellent presentation, ma'am. You have enlightened us with your all knowledge, ma'am. Thank you.
I would to add, I had missed, like I was going through the presentation, there are other drugs that interfere with the calcium metabolism, and they are like tetracycline, quinolones, and thyroids, and a few diuretics also, and the anti-convulsion drugs. So I hope the PG students please add up those points. This point I had missed in the presentation. So please add it to the question, the answers that you write.
Thank you, ma'am. Thank you, ma'am. Ma'am, we have a small appreciation certificate from VISHNU ADHYAAN, ma'am. Please accept, ma'am.
Thank you so much.
Thank you, ma'am. That was a nice lecture from Dr. Vinita Gupta, ma'am. Now we'll have a short break for lunch. We'll have a short break for lunch and we'll regain the session from 1:15. And after the last speaker at 3 o'clock, we have a session by Dr. Dharpan sir to enlighten us on TMJ. Thank you.
VISHNU Dental College and Hospital is in Bhimavaram, West Godavari district of Andhra Pradesh. Established as a teaching institute and service-oriented hospital in 2002, it renders the best quality oral health care to the population in and around the Godavari Delta. The students learn in small groups, enhancing their focus of attention and encouraging proactive participation using e-learning facilities. Well-equipped simulation labs expose the students to practical hand skill proficiency using simulation models and dental mannequins. A wide variety of multi-speciality, inpatient, outpatient, and emergency oral healthcare services are provided with stringent infection control protocols. A state-of-the-art, comprehensive clinical setting facilitates multidisciplinary treatment approach under one roof. Students are exposed to world-class dental instruments, instrument tracking system, digital radiography, and many more. Dental Hospital has employed electronic dental record system for a centralized patient data management. Specialty-wise services rendered include oral medicine and radiology, equipped with digital radiography and the dental CT scan facility to obtain 3D radiographic images. This department also supports 30+ rural dental clinics through teledentistry facility. Oral and maxillofacial surgery section that has advanced surgical care facilities to perform a variety of minor and major oral surgical procedures. A well-equipped 20-bedded inpatient facility includes operation theaters, intensive care unit, and wards. Emergency services are also offered 24/7. Restorative dentistry and endodontics housed with dental operating microscopes for high-quality care anticipating the best possible treatment outcomes. Orthodontics and maxillofacial orthopedics that provides various types of braces for the correction of irregularly placed teeth and also smile correction for all age groups. Department of Pediatric Dentistry provides wide range of dental treatments to children with special emphasis on prevention of oral diseases in a child-friendly ecosystem. It has the facility to treat children with special care needs and highly anxious children under sedation. Periodontology section takes care of tooth supporting structures with advanced surgical and non-surgical approaches. It also provides fine gum surgeries using lasers and surgical microscope. The Department of Prosthodontics rehabilitates patients' lost teeth and damaged facial parts using novel jaw tracking methods, electromyography, and TEC scan to achieve promising reconstructive outcomes. Implantology section performs surgeries with various VISHNU Dental College and Hospital is in Bhimavaram, West Godavari district of Andhra Pradesh. Established as a teaching institute and service-oriented hospital in 2002, it renders the best quality oral health care to the population in and around the Godavari Delta. The students learn in small groups, enhancing their focus of attention and encouraging proactive participation using e-learning facilities. Well-equipped simulation labs expose the students to practical hand skill proficiency using simulation models and dental mannequins. A wide variety of multi-speciality, inpatient, outpatient, and emergency oral healthcare services are provided with stringent infection control protocols. A state-of-the-art, comprehensive clinical setting facilitates multidisciplinary treatment approach under one roof. Students are exposed to world-class dental instruments, instrument tracking system, digital radiography, and many more. Dental Hospital has employed electronic dental record system for a centralized patient data management. Specialty-wise services rendered include oral medicine and radiology, equipped with digital radiography and the dental CT scan facility to obtain 3D radiographic images. This department also supports 30+ rural dental clinics through teledentistry facility. Oral and maxillofacial surgery section that has advanced surgical care facilities to perform a variety of minor and major oral surgical procedures. A well-equipped 20-bedded inpatient facility includes operation theaters, intensive care unit, and wards. Emergency services are also offered 24/7. Restorative dentistry and endodontics housed with dental operating microscopes for high-quality care anticipating the best possible treatment outcomes. Orthodontics and maxillofacial orthopedics that provides various types of braces for the correction of irregularly placed teeth and also smile correction for all age groups. Department of Pediatric Dentistry provides wide range of dental treatments to children with special emphasis on prevention of oral diseases in a child-friendly ecosystem. It has the facility to treat children with special care needs and highly anxious children under sedation. Periodontology section takes care of tooth supporting structures with advanced surgical and non-surgical approaches. It also provides fine gum surgeries using lasers and surgical microscope. The Department of Prosthodontics rehabilitates patients' lost teeth and damaged facial parts using novel jaw tracking methods, electromyography, and tech scan to achieve promising reconstructive outcomes. Implantology section performs surgeries with various implant systems. This section also offers basic and advanced implantology training program for dental graduates. Oral and maxillofacial pathology section supports diagnosing wide array of oral and jaw diseases at cellular level to identify the root cause and treat them appropriately. The department has high-end equipment like the research microscope and the pentahead microscope to facilitate research and academic activities. The institution has a novel proprioceptive derivation clinic which fosters ergonomic principles among students to inculcate working in stress-free postures during early stages of their clinical training. The Central Dental Laboratory is well organized with unidirectional workflow design for fabrication of high-quality dental prostheses. The lab has CAD/CAM facility which includes the laboratory model scanner, 3D printer and a high-end Ivoclar PM7 for 5-axis milling. The institution runs a unique dental technology twinning program in collaboration with the University of Bolton, United Kingdom. The Central Library facility provides unrestricted access to national and international dental journals, books, reviews and magazines with e-borrowing option. A state-of-the-art 3-tier Central Sterile Supply Department CSSD facility is established for reprocessing and uninterrupted supply of sterile instruments to all the clinical areas. World-class instruments from Finland-based company LM Dental are a part of the centralized instrument repository. The RFID technology is used for instrument tracking. VDC employed high-capacity centralized suction system from Germany for efficient liquid biomedical waste disposal and aerosol control in the clinics. The medical center provides 24/7 medical support with a well-equipped clinical laboratory and pharmacy facilities. The outreach wing at VDC actively engages in conducting free community screening and treatment camps using mobile dental unit to reach remote, rural and tribal places. There are various program initiatives developed as per the oral health needs of the area, including free preventive dental treatments to the needy. The service orientation of Vishnu spreads beyond the boundaries to 8 districts of Andhra Pradesh and 1 district of Telangana, with 32 satellite clinics offering basic and expert consultations to the rural areas. In brief, Vishnu Dental College is an epitome of transformation in dental healthcare education and service provision towards realizing their vision, oral healthcare for all, on par with the global standards.
The biggest challenge for any educational institute is to equip the students with practical knowledge and skills effectively. Thus, institutes are particularly emphasizing on curriculum process characterized by approaches to teaching and learning which are both experiential and engaging. VISHNU Dental College, one of the leading dental institutes in India. The institution is making sustained efforts towards consolidation of these state-of-the-art infrastructural facilities for teaching and learning.
As you know, dentistry is both an art and a science, and that's where right from the beginning we were wondering how to bring in technology into dentistry.
We always believe in the motto, strive for excellence. In this process, we update ourselves to the current trends.
In this endeavor, we have partnered with Apple by introducing their iPads and their proprietary educational software in both classroom as well as at patient care.
VISHNU Dental College e-learning team has customized a system which promotes flipped class learning methodology for undergraduate students using their iPads. Students will come prepared with the basic concepts to the class. The professor will facilitate learning by conducting activities and instantly evaluate if the student's learning is adequate and competent.
The Apple iPads had really provided us a great platform for classroom activities. VISHNU Educational Resource for NEET aspirants, we call it as Varna in our VISHNU Dental College, is to provide coaching for the competitive examinations. In this system, we use iPads for conducting online examinations and retrieve the results instantly.
We are utilizing iTunes U, iBooks and other third-party applications from the iPads to gain access to the academic data in the form of lecture keynotes, PDF handouts, supporting videos, images, charts, etc. Apps like BoneBox, Dental Anatomy Mastery are providing us with a 3D or micro scan of all the teeth with a comprehensible explanation.
Although using technology in education is beneficial, it has its share of cons. To curtail any limitations, we have employed a mobile device management solution that monitors any irrelevant usage of the iPads. To facilitate the ease of instructing students, the teachers of VISHNU Dental College are being trained in Apple technology adaptation in a novel approach.
Now it's not just enough for the students to have an iPad, it's extremely important that all the faculty, the 100 faculty we have in our dental college also are on board.
When we are in the clinics, especially the comprehensive clinical setup, we use these iPads to secure the data digitally. And these iPads help us to record the data into our patient management system.
We are probably the only dental college in India which is running a chain of 25 hospitals in the rural parts of Andhra Pradesh, predominantly in the Godavari districts, and we have a plan to take it up to 100 hospitals throughout coastal Andhra Pradesh.
We conduct various community outreach programs in remote areas, and these iPads are very useful in collecting data instantaneously.
In case of difficulty in diagnosis, we connect to our remote management system that connects to our closest satellite clinic doctor.
When you have these number of hospitals, you generate a large volume of data. And one of the things we generate a lot is radiographs. And that's where again we use the iPads, we use the Apple iPads to actually transmit these radiographs through teledentistry to our main hospital. And one new project which is very close to our heart is using artificial intelligence and then analyzing all this data which we are generating from all these clinics to come out with a product which will enable a doctor, mostly in the rural areas and the remote areas, to actually look at those templates to actually diagnose an oral health problem.
I would like to thank Apple for providing all the resources and support in our journey to provide oral health care for all.
Concur and Apple have supported us in taking a leap towards the digitization of our teaching and learning process.
I'm very happy to see our students and faculty all carrying iPads. They go to the classroom, they go to laboratories, they go to the satellite clinics, and we see a lot of efficiency in the system.
VISHNU Dental College and Hospital is in Bhimavaram, West Godavari district of Andhra Pradesh. Established as a teaching institute and service-oriented hospital in 2002, it renders the best quality oral health care to the population in and around the Godavari Delta. The students learn in small groups, enhancing their focus of attention and encouraging proactive participation using e-learning facilities. Well-equipped simulation labs expose the students to practical hand skill proficiency using simulation models and dental mannequins. A wide variety of multi-speciality, inpatient, outpatient, and emergency oral health care services are provided with stringent infection control protocols. A state-of-the-art comprehensive clinical setting facilitates multidisciplinary treatment approach under one roof. Students are exposed to world-class dental instruments, instrument tracking system, digital radiography, and many more. Dental Hospital has employed electronic dental record system for a centralized patient data management. Specialty-wise services rendered include oral medicine and radiology, equipped with digital radiography and the dental CT scan facility to obtain 3D radiographic images. This department also supports 30+ rural dental clinics through teledentistry facility. Oral and maxillofacial surgery section that has advanced surgical care facilities to perform a variety of minor and major oral surgical procedures. A well-equipped 20-bedded inpatient facility includes operation theatres, intensive care unit, and wards. Emergency services are also offered 24/7. Restorative dentistry and endodontics housed with dental operating microscopes for high-quality care, anticipating the best possible treatment outcomes. orthodontics and maxillofacial orthopedics that provides various types of braces for the correction of irregularly placed teeth and also smile correction for all age groups. Department of Pediatric Dentistry provides wide range of dental treatments to children with special emphasis on prevention of oral diseases in a child-friendly ecosystem. It has the facility to treat children with special care needs and highly anxious children under sedation. Periodontology section takes care of tooth supporting structures with advanced surgical and non-surgical approaches. It also provides fine gum surgeries using lasers and surgical microscope. The Department of Prosthodontics rehabilitates patients' lost teeth and damaged facial parts using novel jaw tracking methods, electromyography and TEC scan to achieve promising reconstructive outcomes. Implantology section performs surgeries with various implant systems. This section also offers basic and advanced implantology training program for dental graduates. Oral and maxillofacial pathology section supports diagnosing wide array of oral and jaw diseases at cellular level to identify the root cause and treat them appropriately. The department has high-end equipment like the research microscope and the pentahead microscope to facilitate research and academic activities. The institution has a novel proprioceptive derivation clinic which fosters ergonomic principles among students to inculcate working in stress-free postures during early stages of their clinical training. The Central Dental Laboratory is well organized with unidirectional workflow design for fabrication of high-quality dental prostheses. The lab has CAD/CAM facility which includes the laboratory model scanner, 3D printer and a high-end Ivoclar PM7 for 5-axis milling. The institution runs a unique dental technology twinning program in collaboration with the University of Bolton, United Kingdom. The Central Library facility provides unrestricted access to national and international dental journals, books, reviews and magazines with e-borrowing option. A state-of-the-art 3-tier Central Sterile Supply Department CSSD facility is established for reprocessing and uninterrupted supply of sterile instruments to all the clinical areas. World-class instruments from Finland-based company LM Dental are a part of the centralized instrument repository. The RFID technology used for tracking. VDC employed high-capacity centralized suction system from Germany for efficient liquid biomedical waste disposal and aerosol control in the clinics. The medical center provides 24/7 medical support with a well-equipped clinical laboratory and pharmacy facility. dental healthcare education and service provision towards realizing their vision, oral healthcare for all, on par with the global standards.
Good afternoon, delegates. I kindly inform everyone to join the session. It will start in another 5 minutes. Please tell your friends to join. Very good afternoon, delegates. Now coming to our afternoon session, we have Dr. Shaila Kotwal ma'am's presentation. Ma'am is going to enlighten us on the topic oral defense mechanism. Before going into lecture, I would like to introduce our ma'am. Ma'am has completed her undergraduation and post-graduation from KLE Institute of Dental Sciences under Karnataka University, Dharwad. She has received PhD degree in 2011 from the KLE Academy of Higher Education and Research, Belgaum. In the year 2016, she underwent training in tissue banking and received diploma in tissue banking from the National University of Singapore and has completed a certified course in implantology as well as a course in microsurgery. She has delivered guest lectures in various international and national conferences to her name: amniotic tissue grafts in periodontal defects, allografts and GTR membranes in regeneration of periodontal therapy at the Asian Pacific Association of Surgical Tissue Banks Conference at Malaysia, Korea, Japan, Indonesia, project papers at EASD at Berlin and AAD at Chicago Conference and Indian Society of Periodontology. Ma'am has 58 publications till date in both national and international peer-reviewed index journals, chapter contributions to various textbooks. She is a receiver for editorial board for various scientific journals, and she has contributed by publishing books on local drug delivery in periodontics and herbs in periodontics. She has contributed chapters in books of human amniotic membrane, basic sciences, clinical application, health and disease of oral cavity plaque, microbiology of dental plaque in periodontal health and disease. She has received many awards for her papers and the presentations at various gatherings. Ma'am, I invite you to present and enlighten our delegates with the topic oral defense mechanism. Now session is all yours, ma'am. Thank you.
So today we are just going to brush up the topic oral defense mechanism as a revision program for the postgraduates. So to introduce this particular topic to you, like the oral cavity has been designed in such a way that like the structures which are present also help in the self-cleansing mechanism. For example, the caul, the vestibule, the cheek mucosa, and even the tongue. So they all help in the self-cleansing activity. So whether to remove the initial accumulation of the plaque, or that's a pellicle, or even the food particles. It's mainly the food particles. What I want to stress is like. So apart from that, when we gingival fibers, when we talk, these particular gingival fibers braces the marginal gingiva firmly against the tooth surface, and they provide rigidity to combat the forces of mastication and tends to throw away the foreign materials which are into the sulcus. The expulsion is aided by the movement of the teeth and the gingival tissues during the mastications. So what is the oral mucosa participation in the defense mechanisms? So the oral mucosa, that is the oral cavity. So when we consider the oral cavity is the entry of the GIT tract participating in the mastication and the tasting the food. Therefore, the structures and the anatomy of the oral cavity are quite unique. The oral mucosa is a membranous tissue lining inside the mouth consisting of oral epithelial cells and the layer of the connective tissue underneath known as the lamina propria. Unlike the gut mucosa, which includes a single layer of columnar epithelial cell, the oral mucosa comprises of stratified epithelium consisting of increasingly catenaced layers of squamous epithelial cells piling over the lamina propria. The catenation degree is not homogeneous throughout the oral mucosa. It varies depending upon the anatomical locations and functions. For example, the masticatory epithelium on the palate, which could be easily injured through the chewing, is protected by the thick keratin layer resembling the skin, which is likely to make antigen entry difficult. On the other way, or other sides, such as the lining mucosa, that's the internal part of the cheek inside the lips, floor of the mouth or sublingual area, features no keratin layer. So that means the degree of keratinization helps not only to resist the antigen entry, but also by resisting the antigen entry, they try to also prevent the inflammation entering into the tissues. So for example, when we talk about the attached gingiva, we always say that like the one of the function of the attached gingiva, since the epithelium is keratinized into it, like, so it helps in preventing the inflammation. So this particular epithelium and other important function of this particular epithelium has got this exfoliation of the cells of its outer layer, which is continuously happening and is continuously getting renewed. So when you look at a particular slide, so we can see the different functions of this particular cellular elements of the epitheliums. So now let's take it the first is the cell renewal. Because of the continuous cell renewal which is happening, the tissue turnover takes up. And because of cell migrations and reepithelialization helps in the wound healing. The third aspect of this is like, okay, the cell attachments and the tissue integrity of the cellular elements of this epithelium. So this will help in the toughening and the mechanically resistant surface. The presence of the antimicrobial peptide in this epithelium, so it indicates the early responders of the innate immunity. So when we look at these particular pictures, so we can see the keratin layers and the cells of the keratin, they're quite closely spaced. And in the second slide, so where it's only the lining mucosa, so we can see the cellular elements, how sparsely spaced, and also the antigen entry here. Okay, so this indicates the different type of the epithelium at the presence of the palate to palate, and this is the area where we can see the lining mucosa. So here it's showing this particular slide is showing the entry of the antigen. So where the dendritic cells, that's the Langerhans cells, which are also called as the antigen presenting cells, they capture the first antigen and they start the other events later. So when this particular antigen, as we see in this particular picture also, like the antigens entering, okay, so into the oral cavity and so ending into the tonsil directly, they are showing the picture of the tonsils here. Okay, so but here when we look at this particular slide, this antigens captured after this particular events from the epithelium, getting into the recognizing by the dendritic cells, and they get opsonized, or even if it's present like, okay, they get entry into the directly the lymphoid follicles. So the lymphoid follicles is a single follicle which is shown by this particular picture. These are the cells of the gingiva which are participating in the defense mechanisms. We all know this for all the cells, the neutrophils to monocytes, basophils, eosinophils. So they are also present normally, and they start participating in the event by releasing their enzymes as the inflammation initiates and progresses. These are the non-immune cells, that is the keratinocytes, Langerhans, and the fibroblasts, which we'll be talking later. So now let's talk about something about saliva. So what is the saliva? Saliva is a watery fluid secreted into the mouth by the salivary glands that functions in tasting, chewing, and swallowing food, moistens the mouth, and starts the digestion of the starch. And these salivary glands, or this saliva, is secreted by the different glands that is a parotid, submaxillary, submandibular, and sublingual. So collectively, the secretion of this particular glands collectively gets into the oral cavity as a saliva, and the components of the saliva are produced by the serous and the mucous acinar cells within the gland, and it gets drained into the oral cavity. So we may find the different components into it also like. So now let's see what are the functions of the saliva. Yeah, this whole complete circle shows the functions of saliva and the associated enzymes along with it. For example, the taste. So when we eat something like, yeah, definitely the taste buds are there, like we can taste the food. So what is going to help in tasting the food is the carbonic anhydride, zinc, and the water. So that gives the, along with the taste buds, that gives the food taste to the any person. So then the bolus formation. Bolus formation is favored with the help of the mucins and the water. So whenever we have the reduced salivary formation, yeah, definitely we find the difficulty in the bolus formation. And so even that particular reduced saliva also, which is termed as a xerostomia, lands into the different types of the anomalies or the problems. So once the bolus formation is there, like, okay, we are definitely it is going to be digest. So the enzymes which are going to help in the digestion are the amylase, protease, lipase, DNase, or RNase. These are enzymes. So this is the food. So which completes the helping in the tasting, bolus formation, and the digestion of the food into the GIT. So apart from this, like the saliva also helps in the buffering, that is maintaining the pH of the oral cavity. So this is favored by the bicarbonate phosphate proteins. So this is a particular mechanism which helps or maintains into the oral cavity, so that like the pH is always maintained in the oral cavity. So whenever there is an alteration in this pH to the, so definitely we have the shift in the pH, because when there's a shift in the pH, like we definitely will have the caries, increase in the caries formation or the growth of the anaerobic microbiota. So you might have already seen that particular clinical aspects in the oral cavity. So the protection against the demineralization. So the mucins, the calcium, and the phosphates, so which help in the protection against the demineralization, particularly with the procedures. The remineralization is favored with the help of the saccharin, calcium phosphate, and PRPs. The lubrication, the mucins and the PRGs. So other functions like antibacterial, antifungal, antiviral are also carried out by the different types of the enzymes. So which we are going to discuss individually with the particular enzymes. So now let's take up the saliva and its functions, which is going to carry out at the different levels. So slightly, we'll just go in detail about this. So first it is saliva microbe interactions. So we know commensals microbiota are present in the oral cavity and are non-pathogenic. The shift of this microbiota takes place when the plaque starts accumulating on the tooth surface. So saliva helps in preventing the plaque formation by forming the aqueous pellicle and also preventing hydroxylated surface from the acid attack, thus inhibiting the demineralization of the tooth surface, which we saw in the previous slide also. So other important function of salivary molecules include buffering of acids and neutralizing the toxic products produced by the microorganisms. Again, which was again I specified in this previous the chart. So when we talk about the clearance of the bacteria from the oral cavity, so that is always favored with the formation of the agglutinins. So it has been shown in vitro experiments, many specific of bacteria gets crumpled or aggregated. So when suspended in saliva, so it is thought this aggregation in vivo helps in promoting the clearing this particular microbiota from the oral cavity. So apart from this, binding of salivary components to the bacterial surface may also prevent the adhesion of the bacteria to the tooth surface, to the mucosa and the plaque. So other components of saliva that have been shown to agglutinate bacteria includes the mucin, salivary IgA and the palatal agglutins. And these salivary mucins are nothing but a large molecular weight of glycoproteins containing 30 to 80% of the carbohydrate and are secreted by mucins SNR cells or salivary glands. There are many molecular and genetic studies which have clarified the chemical nature of these mucins and identified the additional types like mucin 1, mucin 4 and gel forming mucin 5B. So in the oral cavity. So apart from this, like there are several other mucins are also been identified in the different parts of the body. So mainly we are concerned about this in the oral cavity, the above mucins. The higher molecular weight of the salivary glycoproteins, including the mucins, can aggregate the oral bacteria and facilitate the clearance from the oral cavity. So when we now let's discuss something about the antimicrobial component saliva. So mainly the components which participate in this antimicrobial aspect of saliva is the lysozyme, lactoferrin, myeloperoxidase, salivary cystatins, defensins, salivary immunoglobulins, and phagocytic cells, and so on. Like few more are there. Like so I have counted only the most important aspect of it. Like so. Now let's get into the little bit of detail about this particular enzymes, how they are going to act. Similar to the, so now when we talk about the lysozyme, so lysozyme is a secretion which is found like how the secretions of found in the nasal fluid, seminal fluid, the breast milk. So this is also found in the saliva. And several salivary components have been shown to kill or inhibit the growth of the bacteria. So this lysozyme is one of the first salivary antimicrobial component to be described, and it is a 14 kDa protein that hydrolyzes the beta-1,4 glycosidic linkages between N-acetyluric acid and N-acetyl glucosamine in the peptidoglycans of the bacterial cell wall. The resultant weakening of the cell wall is susceptible to bacterial species, which results in the lysis of the bacteria. So other function is it also involves in the activation of the endogenous bacterial enzymes that are involved in the cell wall metabolism and can cause the lysis of the bacteria that are insensitive to mucosamidase activity of lysozyme. Secondly, the lysozyme can also bind to the oral bacteria and aggregate them, so helping in the or facilitating the clearance from the oral cavity. So next one is the lysozyme. We completed now. So now we'll talk about the lactoferrin. So lactoferrin is a glycoprotein of 75 kDa synthesized by glandular acinar and epithelial cells as well as the inflammatory cells. Lactoferrin binds 2 atoms of iron per molecule with a simultaneous binding of 2 molecules of carbonate. The primary function of lactoferrin may thus be bind to sequester iron from the bacteria. So in addition, iron-free lactoferrin may possess a direct iron-dependent bactericidal effect on various strains of bacteria. For example, it may be the P. gingivalis. So now when we talk, now next let's come back to the myeloperoxidase. Myeloperoxidase, which is found in saliva, has its origin in polymorphonuclear cells, which are found in the substantial number in the oral cavity. These cells migrate from the gingival crevice and through the oral mucosa. Leukocytes are rich in myeloperoxidase and lyse cells release active enzyme into the saliva. Myeloperoxidase is an abundant protein making up to 5% of the total protein. It might utilize hydrogen peroxidase to generate toxic oxidized halide derivatives, or it might play an important protective role against the oxygen-free radicals. So next we'll talk something about the salivary peroxidase. Salivary peroxidase consists of peroxidase enzyme, the thiocyanate ion, and hydrogen peroxide. The enzyme found in secretion such as tears, milk, and saliva, and is able to inhibit bacterial growth. And it catalyzes the oxidation of SEN by H2O2, generating highly reactive oxidized forms of thiocyanate such as OSEN. These products have direct toxic action on a variety of microorganisms. Salivary peroxidase can also neutralize the deleterious effect of hydrogen peroxide produced by a number of oral microorganisms, which reduce the acid production by glucose stimulated dental plaque and inhibit the glucose uptake by the microbiota. Salivary peroxidase system also interacts with the other defense factors such as the lysozyme lactoferrin, and it has also been demonstrated that peroxidase system can also bind to the IgA, IgM, IgG antibodies, but the combination of the IgA peroxidase has more enhanced than other combinations. So other enzymes like histatins. Histatins are the family of small basic peptides characterized by the high content of histidine. At least 7 members, one of which is a phosphorylase, have been identified in a human saliva. These vary in size from 3 to 5 kDa. These molecules, which are present in aqueous enamel pellicle and inhibit the precipitation of the calcium phosphate salts, which has been shown recently to display the bactericidal and the fungicidal activities. Histatins are secreted mainly in parotid and to the lesser extent by submandibular saliva. There are 12 salivary histatins which has been isolated from saliva, and they possess the antimicrobial properties against the few strains of the Streptococcus mutans and inhibit the heme agglutination and periopathogens like Porphyria gingivalis. In addition to this, histatins also neutralize the endotoxic lipopolysaccharides which are located in the outer membrane of the gram-negative bacteria. So apart from this, they also are potent inhibitors of the growth and germinations of the Candida albicans, and also have the similar activity like synthetic antibiotics like imidazole. So now let's come to the defensins. These defensins are a subfamily of the homologous antimicrobial peptides constituting of the important component of the innate immunity. They are found predominantly in the vertebrates, and they are among the proteins expressed at the highest level of the oral mucosa. They are present in the oral inflammatory conditions, oral carcinomas, and some cell lines derived from the oral carcinomas also. And salivary immunoglobulins, we all know that. So they are mainly of the IgA, IgM, and IgG. So where salivary immunoglobulins is a predominating one is the IgA. So we'll be talking about this IgA participation in the several mechanisms. The last one which we are going to talk is about the phagocytes. So these phagocytic cells are present in the bloodstream and in the tissues, which are capable of engulfing and digesting the foreign material. The two most important phagocytic cells are polymorphonucleoleucocytes and the macrophages. The polymorphonucleoleucocytes protects the body against the acute invasion and has the ability to amoeboid the movement and can pass through capillaries and through the tissues, including the gingival connective tissue and epithelium. The direction of their movement is determined by the tissue damaged products, which are chemotactic in nature. And the macrophages are the cells which start the life as a monocyte and swells in the tissue to become an efficient phagocyte which is capable of digesting the large foreign particles. The macrophages also take up the antigens in the circulating fluid for presentation to the lymphocytes. Phagocytosis is also aided by a battery of 9 related proteins, which are known as the complements. So, which act by immobilizing the bacteria or toxins, so that the phagocytes can act more effectively in disposing the foreign matter. The saliva also has a role as an antiviral. Saliva also, which has got many antiviral factors. For example, the infectivity of HIV has been shown to be significantly suppressed in the presence of the human saliva, and antiviral factors are also oscillated from the saliva. So saliva also, it has got so many other functions, additional functions. So what we have is salivary antioxidant system. Salivary antioxidant system is an essential in possessing the anti-carcinogenic role in the oral cavity, which is aimed at fighting the reactive oxygen species and reactive nitrogen species, which are caused by the smoking, alcoholic beverages, food, coveted drinks, dental restorations, and various other volatile sources which are freely entering the oral cavity through the body's largest open gate, that's the mouth. The salivary antioxidant system includes various molecules and enzymes. The most important are the uric acid molecule and the peroxidase enzyme. The both are water soluble. Uric acid contributes approximately 70% of the total salivary antioxidant capacity. It has been shown in the animal model that anti-carcinogenic capability of saliva significantly inhibits the initiation and progression of the oral cancer. And in a study done using the MS test, it has showed saliva inhibits the mutagenicity of oral cancer inducers, that is cigarette smoke and 4-nitroquinone oxide. Saliva also plays an important role in preventing cigarette-induced deoxyribonucleic damage. This antioxidant capacity of saliva protects against the oral cancers. However, in cases of periodontal disease, the formation of and progression of the normal cellular mechanisms are hampered. The oxidation process occurs due to the increased reactive oxygen species reduction induced by the etiological factors of periodontitis, such as the bacterial toxins from the plaque formation. And salivary buffers and coagulation, when we speak about this salivary buffers, we have already specified in the previous slides. Okay, coagulation factors. The saliva also contains the coagulation factors as 8, 10, 9, and the plasma thromboprotein and the Hegman factor, which hasten the blood coagulation and protect the wounds from the bacterial invasion. So the other enzyme like the catalytic enzyme, which is also present in favors this in the coagulation aspects. Now let's talk something about the gingival clavicular fluid. So gingival clavicular fluid, as we know, like it's a which is present in the gingival sulcus. So many of us are still many times like were in the confusion, whether it's the transudate or exudate. So, and finally we have come to it has been come to the conclusion and errors like, okay, so now what is happening during the inflammation is an exudate, or in the healthy condition is a serum transudate. So what is this transudate is like? Transudate is result of the increased pressure in the veins and capillaries that forces the interstitial fluid through the vessel walls into the crevice. And the transudate is a protein for watery interstitial fluid, whereas the exudate contains the higher content of the protein, including the major plasma proteins and immunologically active components. So this exudate, or what we call as inflammatory exudate, so when it starts appearing, so it will start appearing so which will be discussed in the later slides. So before that, so let's have a view what does GCF has its function to play as a in the defense mechanisms. So firstly, it prevents the entry of the bacteria within the sulcus and flushes away the bacteria, and it has got a defensive proteins in it, and it maintains the structure of the epithelium and acts as an antibacterial activity with the presence of the molecules like myeloperoxidase. Apart from that, like during the inflammatory process, like it has got a complements into it, like, and plus it has got additional, which is very important factors are the presence of the immunoglobulins, which is predominantly present as the IgG. So whenever there is an inflammation, there is a vascular phase of during the vascular phase of inflammation, the microcirculation gets affected, resulting in the increased leakage of the fluid from the gingival capillary beds and the influx of the polymorphonuclear leukocytes, which creates a cell-rich inflammatory exudate. Production of this inflammatory exudate is a hallmark of the acute inflammation or inflammatory response to provide the primary non-specific defense. And so this will result in the formation of the many host-derived products, which includes the histamines, leukotrienes, heparin, serotonin, hyaluronic acid, as well as the antimicrobial enzymes factors. So this particular slide shows, so during this particular event of the inflammation, so we get the host-derived enzymes and their inhibitors, and the tissue breakdown products, and the inflammatory mediators, and the host response modifiers during the process of the inflammation. So all these particular products have been used as a GCF markers or for detecting the various forms of the destruction of the tissues. So there are many studies or the many researchers have been gone into a particular studies like, okay, so and they have validated these particular GCF biomarkers which have caused the initiation and the inhibition of the pathological process. And so when we look into the study carried out by the Barros et al., this team indicated that the GCF is a source of multiple biomarkers that potentially indicates the disease presence with the details of active and the passive sites. So in another study, so which has provided a comprehensive overview of the host defense mediators and their expressions in the clavicular fluid around the tooth, and these studies favored the use of the GCF as a universal tool. And also this particular GCF markers have been used even for many to study the many systemic conditions also like. So mainly what I can say is the GCF definitely clavicular fluid is a transudate. So during and plus like it is composed of the many cellular organic and inorganic elements along with this particular the factors which will help in the defense mechanisms what we discussed earlier. So but during the inflammation, so as the inflammation starts or during the inflammation, so we have got, as we discussed, many metabolic products and many post-derived products which we get and which will indicate the significance of the which particular tissue has undergoing the destructions. So here in this slide we can see, so these are the particular toxins. So when we talk about the enzyme and enzyme inhibitors or the host-derived products or the metabolic products, so this all indicates which, depending upon which is the particular enzyme and which is a particular metabolic product, from where the source is there and what is its action and what is the correlation of this particular disease activity is due to. So for example, like we have taken this acid phosphatase, sulfonic phosphatase, pyrophosphatase, the source actions and the correlation. So always as to the postgraduates, so you can depending upon the time you can just pick up the particular the events which are happening and select the particular enzymes which are very important, and you can explain them. So instead of like going in detail with each and every aspects. So similarly, even you have the lysozyme, lactic dehydrogenase, so where this particular slide shows the source, actions, and correlation. Like for example, like lactic dehydrogenase is there, like it version is from the bacteria, and actions, it catalyzes the conversion of the periodic to lactate. But there's no significant correlation according to the many studies have been done like. But the lysozyme correlates with the positively with the severe periodontal destruction. So where we can even the stages have the like mild, moderate, and severe, we can come to the conclusion. So similarly, like okay, beta glucuronidase is there, like where we can so correlate with the positive correlation with the GSF flow and the pocket depth. So definitely this particular all these events with this breakdown products will help us to understand which tissue or the which tissue of these particular surrounding tissues have been affected. So which directly we can signify that, so that like we can come out with the particular tools which can help us on the chair side, which is already existing, and we can still come up with many more. So with all this, whether it's a GCF, saliva, or the epithelial barrier, these are all have got a particular, we understood the cellular elements and the many enzymes are present, like which will take care, and even the structures of the oral cavities is designed in such a way that like they try to take care of this particular antigens entering into the body. But in spite of all this, like so many a times like the antigens enter into the body, it's like okay, so like our host gets initiated with the host response, and as the innate immunity gets into the actions. So let's watch this particular video, so where we can understand the basic events of the
immune
immunity.
system is the body's defense system. It protects the body from disease-causing organisms called pathogens. The protection has several layers. First, invading pathogens meet with a number of surface barriers, which consist of physical, chemical, and biological obstacles designed to keep them out. The primary physical barrier is the skin, which covers the entire body. Body systems that are open to outside environments such as the respiratory, digestive, urinary, and reproductive system each have their own mechanisms to prevent entrance of microbes. Mucous membranes trap them, sneezing or coughing reflex expels them, while urine mechanically flushes them out. Chemical barriers include stomach acid and various antimicrobial substances in sweat, saliva, tears, and other body fluids. The skin and mucous membranes are also heavily inhabited by the body's normal flora, which competes with pathogens for nutrition and space, providing biological barriers. If an organism manages to get past the surface barriers, for example, via a splinter that pierces through the skin, it will meet with the innate component of the immune system, which mounts an immediate but non-specific response. If this fails to contain the infection, another layer of defense, called the adaptive or acquired immune response, comes into play. The adaptive response takes longer to be activated, but is more effective as it specifically targets the invading pathogen. It also leaves the body with a memory of the pathogen, so it can react faster the next time the same pathogen attacks. The major players of the immune system are the white blood cells, or leukocytes. All leukocytes derive from hematopoietic stem cells in the bone marrow. Each of them has different roles in the immune response. The first response of the innate immune system is inflammation. Resident macrophages, which constantly patrol body tissues, ingest the pathogen and release inflammatory chemicals called cytokines, which attract other immune cells to the site of injury. Basophils, eosinophils, and mast cells release their own cytokines, amplifying inflammation. Cytokines dilate blood vessels, increasing blood flow, and are responsible for clinical signs of inflammation, such as redness and swelling. They act on endothelial cells of blood vessels and serve as chemical cues for migration of neutrophils, the major phagocytes involved in first-line defense. Activated endothelial cells attach to neutrophils in the flow, slowing them down before getting them to squeeze through the vessel wall. Neutrophils engulf bacteria and destroy them with enzymes or toxic peroxides. They may also release highly reactive oxygen species in a phenomenon known as oxidative burst, which kills pathogens faster and more efficiently. The neutrophils themselves, however, also die in the process, their debris forming pus on the injury site. The adaptive immune response starts with the so-called antigen-presenting cells, of which dendritic cells are most effective. Resident dendritic cells on the site of infection swallow up pathogens, cut them into pieces, called antigens, and display them on their surface. These dendritic cells are then picked up by lymphatic capillaries and travel to lymph nodes, where they present the antigens to a matching T cell. The pathogen itself may also travel to a lymph node where it may encounter a matching B cell. The match-finding process underlies the specificity of adaptive immune response. T cells and B cells exist in billions of variations. Each carries a unique surface protein, which acts like a key. Among these billions of keys, only the ones that can bind to or unlock the invading pathogen are activated. Activated T cells and B cells undergo differentiation and proliferation, called clonal expansion. This process produces memory cells, ready for future infections by the same pathogen, and effector cells, which include activated cytotoxic T cells and plasma B cells producing antibodies. Both of these are specific to the pathogen. Antibodies and cytotoxic T cells then leave the lymph node for the bloodstream to be delivered to the site of infection. Antibodies attach to pathogens and either target them for destruction or neutralize them. Cytotoxic T cells release toxins to kill infected host cells. Thank you for watching. If you like our videos, be sure to
of the invading micro of the invading micro how the antigen enters in. So finally, I understood how the antigen enters into the body and how it is taken care by the host, the different immune mechanisms. So finally, what is required is like, okay, so the destruction of the invading microorganisms. It can be either through the innate immunity or the acquired immunity. And finally, with all these particular classifications, what we have understood till now, now, with this, like what we discussed till now, the oral structures, the saliva, GCF, and the video presentations of this immune host response with the immunity, like, okay, so there are the different mechanisms of the, whether it's the innate immunity or the acquired immunity. So let's conclude that, like, okay, so the oral structures and the host response and this particular saliva and GCF has helped to defend the oral cavity against the microorganisms, and the oral cavity is well equipped with the counteracts of the adverse conditions which may harm the gingiva, while the innate immunity acts primarily against the foreign invader, and specific immunity takes more complex targeted approach to protect gingiva. So right from the superficial epithelial layer to the innermost connective tissue, there's a line of defense that acts harmony with the other oral structures and maintains the homeostasis. So I have made the subject clear to you. Thank you everyone and everyone.
Thank you, ma'am, for the wonderful lecture. It was a great tap into your knowledge, ma'am. You have just enlightened us regarding the topic. Ma'am, over to you.
Hello, Shaila. That was a very nice presentation. There are so many lines of defense in the body, and that is so pragmatically created by the creator. And you dissected it nicely into various, portions and made us understand. And thank you so much. You have been always a very active and very much involved in the, research, and also all your research knowledge has been put in here, and it has definitely benefited everyone. Thank you. Thank you so much.
Thank you, ma'am. It was lovely having your presentation on the screen, although you are not able to make it on live because of your busy schedule. It was a wonderful presentation, and thank you for accepting our invitation and readily you are there to share your knowledge from the examination point of view, which was really awesome. Thank you so much, ma'am.
Thank you so much, ma'am. We have a small token of appreciation from Vishnu Adhyaan. All the questions will be answered through mail. Dear delegates, it's a time for our last lecture in this session. Before I introduce our ma'am, Dr. Mala Dikshit ma'am is a professor and head in the Department of Periodontics from Nair Hospital, Dental College. So madam is going to enlighten us on the role of vitamins and minerals in health and disease. Dr. Mala Dikshit ma'am joined the School of Dentistry in year 1984 at the prestigious college GDC, Mumbai. She completed her undergraduate and postgraduate studies in 1993 under the guidance of Dr. R.H. Jaffer and S.A. Kale. And she served her alma mater for 5 years, where she learned the basics of academic teaching. And she then moved on to an associate professor at Padma Shri D.Y. Patil Dental College, an upcoming college in those times. In 1998, being a junior most, did all the jobs and saw the college becoming from 40 seat to 100 seat intake capacity. During this process, she rose to the post of professor and HOD. This helped her in learning the administrative and clinical skills. Ma'am has an opportunity. She had joined an opportunity in NHDC as a head in the Department of Periodontics through the MPS selections in 2005. And she has been a part of many universities as an examiner for both PG, UG and PhD examinations. She has been actively publishing papers in various national and international journals, also doing yeoman services to the public at large by conducting camps, distributing educational handouts, penning out an informative, authentic, scientific-based tidbits in common magazines. She has been in Nair Dental Hospital College for 14 years, rendering her services and commitment towards the college, students and patients as well. We welcome you, ma'am, all All of us are eager to hear your words and presentation on role of vitamins and minerals in health and disease. Over to you, ma'am.
Hello, good afternoon, everyone. I request you to kindly share the recording sent to you. Hello, can you hear me?
Yes, ma'am, we can hear you.
Yeah. Going to kindly share the PPT which I sent to you, the recording.
Okay, ma'am.
Or should I share? Yeah. Good evening, everyone. With this presentation, we come to the end of this convention. Greetings from Nair Hospital Dental College, Mumbai. Mumbai, the city known for its star-studded hotels, the Worli ceiling, the Queen's necklace, vada pav, which is a full meal for many but has got no vitamins and nutrition. This presentation is brought to you from Nair Hospital Dental College, which is situated in the heart of the city that never sleeps, which comes under the Municipal Corporation of Greater Mumbai. The role of vitamins and minerals in health and disease is the topic that I am given to engage you for the coming next 50 minutes. I will take you through the introduction, the nutrients, vitamins A, D, E, K, B, C, minerals and trace elements, why does deficiency occur, nutrition and periodontal interrelationship, nutrition and periodontal disease, and we shall conclude with some thoughts. Vitamins and minerals are micronutrients required by the body to carry out a range of normal functions. However, these micronutrients are not produced in our bodies and must be derived from the food that we eat. Nutrients can be macronutrients like carbohydrates, proteins, and fats, and micronutrients like vitamins and minerals. Micronutrients needed by the body are in very small amount. However, their impact on the body's health are critical, and deficiency in any of them can cause severe and even life-threatening conditions. They perform a range of functions, including enabling the body to produce enzymes, hormones, and other substances needed for normal growth and development. Vitamins are a group of organic compounds which are essential for normal growth and nutrition and are required in small quantities in the diet because they cannot be synthesized by the body. They can be classified as oil solubility and water solubility. Fat soluble vitamins are vitamin A, vitamin D, vitamin E, vitamin K. Vitamin D can further be divided into vitamin D2 and D3. Vitamin K can be further divided into vitamin K1 and K3. Water soluble vitamins are B complex vitamins and non-B complex vitamins. Vitamin B can be again divided into thermolabile and thermostable. Thermolabile is vitamin B1 and thermostable ones are vitamin B2, B5, B6, B12, niacin, and folic acid, and vitamin H, and non-B complex vitamin is vitamin C. Fat-soluble vitamins, let's find out the functions in metabolism, the symptoms during deficiency, and the whole food, the sources that we can get it from. Vitamin A, the dietary recommended allowances is given as in the chart on the right. Vitamin A helps in cell growth and development. It supports vision. It supports the immune system and helps in healthy development of the fetus. They are basically carotenoids, the pigments that are present in fresh fruits and vegetables. Basically, three major ones: alpha carotene, beta carotene, and beta cryptoxanthin. They're all pre-retinoid activity makers, that is, they help in formation of retinoids. Others like lutein, lycopene, zeaxanthin, astaxanthin, and canthaxanthin. Lutein and zeaxanthin found in green vegetables like spinach and kale. Lycopene found in fruits and vegetables with red and orange color. They help in release of anti-inflammatory cytokine interleukin-10, and reduction of TNF-alpha. Lycopene, it's most effective as an oxygen quencher. Its deficiency in the body can lead to dry skin, dry eyes, night blindness, infertility and trouble in conceiving, poor wound healing, acne and breakouts, delayed growth, throat and chest infections. Its oral manifestations is seen as enamel hyperplasia, xerostomia, increased invasion of periodontium by oral microbe, and increased susceptibility to caries. Papillon-Lefevre syndrome, if treated with retinoid therapy, shows improvement in dermatological manifestations. Doggington et al. Proved that a higher dietary intake of beta-carotene significantly lower percentage of sites more than 3 mm pocket depth at a follow-up after a non-surgical periodontal treatment. Synergistic effect of carotenoids are more effective than single compounds in preventing oxidative Vitamin D. The best source of vitamin D is from sunlight. The recommended doses are given in the chart on the right. Vitamin D helps in bone and teeth development. It helps in muscle contraction, in nerve conduction, and supports our immune system. The symptoms that is seen on vitamin D deficiency in our body is fatigue, digestive problems, immune impairment, joint pains, dry skin, high blood pressure, mood swings, overweight, and excessive sweating. Its oral manifestations can be seen as enamel and dentine calcification defects, large pulp chambers, delayed eruptions, and increased bone resorption. Vitamin D intake has been reported to have many benefits in maintaining periodontal health due to its direct role in bone metabolism, antibiotic effects against periodontal pathogens, and ability to interrupt inflammatory mediators that induce periodontal tissue destructions. One study based on the US National Health and Nutritional Examination Survey database reported that both men and women aged more than 50 years in lower quintile of serum 25 hydroxy vitamin D had higher periodontal attachment loss than those in the highest quintile. Literature supports that periodontitis is associated with vitamin D serum levels. However, an adjunct of non-surgical periodontal therapy remains unclear due to its shortage of available studies. Vitamin E, the recommended dosage is as given in the chart. It helps in protecting the cells against free radical damage, helps in nerve and bone health. It's an anticoagulant. Normal functions of the brain is continued with vitamin E, supports immune system. The symptoms that is seen in vitamin E deficiency in our body is low visibility, fertility problems, immune impairment, leg cramps, dry skin, muscle weakness, anemia, hair loss, and cancer. Oral manifestation of vitamin D deficiency leads to increased bone loss, increased oxidative damage to tissues. Clinical indices and decrease the amount of attachment loss as given by Beffarnia et al. In 2021. Vitamin K. The required recommended dosage of allowance is as given in the chart on the right. It helps in blood clotting, helps formation and maintenance of bone structure. It protects the immune system. Its deficiency can lead to blood clots underneath the nails. It leads to bleeding seen underneath the mucous membranes. Easily bruises are often seen. Stools can be seen in the blood. Oral manifestation shows gingival bleeding, petechiae, purpura, and ecchymosis, and spontaneous bleeding. In 1998, Rawalson et al. Performed cross-sectional studies investigating health and disease sites from 18 patients with adult periodontitis aged between 27 and 64 years. The study result reveals that levels of phylloquinone in GCF lower in health than disease sites. Since phylloquinone is an absolute growth requirement for black pigmented anaerobes, many of which are implicated in periodontal disease, vitamin K at disease sites provide nutritional requirement favoring the growth of black pigmented anaerobes. In 2007, Hojo et al. Assessed the possibility that bifidobacteria compete with P. gingivalis for the mutual growth factor vitamin K. He concluded that bifidobacterium decreased vitamin K concentration and inhibits the growth of P. gingivalis by competing for the growth factor in the environment. How do we manage patients with bleeding disorders and periodontal problems? We have to take a detailed case history of the drugs, the past episodes, and the previous hospitalization history of the patient. Investigations like prothrombin time, APTT, INR, bleeding time, clotting time, and risk assessment needs to be noted. Intraoperatively, professional cleaning and scaling and root cleaning can be safely performed with the use of local antifibrinolytic mouthwashes such as tranexamic acid or epsilon aminocapric acid. Regional block anesthesia must be avoided. Local infiltration anesthesia with the use of vasoconstrictive agent is desirable. Lidocaine 2% with 1 in 1 lakh epinephrine is adequate. Articaine 4% with 1 in 1 lakh epinephrine also provides sufficient anesthesia for surgery in the mandible. Post-operative care should include high protein diet, lots of liquids, and use of mouthwash. Water-soluble vitamins. Let's find out the functions in metabolism, symptoms in disease in deficiency, and whole food sources from where it can be obtained. Vitamin B. The benefits of vitamin B complex, referred to as vitamin B complex because there are 8 vitamins: vitamin B1, B2, B3, B5, B6, B7, B9, and B12, play an important role in keeping our bodies running like well-oiled machines. These essential nutrients help convert our food into fuel, allowing us to stay energized throughout the day. B9 helps cell makes and maintain DNA. B1 and B2 are important for healthy functioning of the muscles, nerves, and heart. B6 supports the immune system and aids the body in breaking down proteins. B3 helps regulate the nervous and digestive system. B5 and B12 are required for normal growth and development. B7 is involved in the production of hormones. Now, this is a chart showing the different dietary requirements and the sources where it's available from. B1, that is thiamine, it leads to deficiency, leads to fatigue, nerve and brain damage, and leads to beriberi. It is found in dietary sources like cereals, spirulina, beans, lentils, flax seeds, and other seeds, milk, pork, nuts, oats, beef, rice, and wheat. And the main functions are to convert the protein, carbohydrates, and fat from food into energy, nervous system functions, and synthesis of DNA. B2, that is riboflavin, it creates deficiency into cracks in the lips, tongue swelling, and other skin issues, leads to arabinoflavinosis. It is usually found in milk, dietary products, eggs, fish, green leafy vegetables, cereals, whole grains, and liver. The main functions being to convert protein, carbs, and fat from food into energy. Skin health and eye health is an important function. B3, that is niacin, leads to deficiency into muscular weakness, loss of appetite, and leads to pellagra. Found in yeast, meat, fish, milk, and dairy products, seeds, eggs, green vegetables, beans, cereals, and grains, nuts, and poultry products. Its main function is to convert protein, carbohydrates, and fat from food into energy, fatty acid synthesis, and improves the cholesterol. B5, that is pantothenic acid, no determined deficiency found as yet, found in fresh meat, vegetables, and unprocessed grains. It helps in converting carbohydrates, fats from food into energy. production of red blood cells, healthy digestion, and hormone production. B6 pyridoxine leads to muscle weakness, anxiety, and depression in deficiency states, usually found in liver, meat, fish, poultry, wheat grams, bananas, beans, and legumes, and its main function being protein and amino acid metabolism, release of stored glucose, hormone production, and helps in brain function. B7, that is biotin, its deficiency leads to loss of appetite, vomiting, and depression, found in eggs, nuts, fish, butter, beans, whole grains, cauliflower, bananas, and mushrooms. Main function is to convert protein into energy, carbohydrates metabolism, fat synthesis, hair and nail health. Vitamin B9, also called as folic acid, leads to GI pains and anemia, found in grains, bread, vegetables, cereals, bananas, oranges, beans. Its main function is to protein and amino acid metabolism, DNA synthesis, and formation of red blood cells. B12, that is cobalamin, leads to nerve damage, fatigue, and anemia in case of deficiency, found in liver, yogurt, dietary products, fish, clams, oysters, non-fat dry milk, salmon, and sardines. Main function being food metabolism, energy production, DNA synthesis, formation of red blood cells, brain, and nervous system functions. Oral manifestations of vitamin B deficiency is thiamine deficiency, can be called as beriberi, minute vesicles simulating the herpes on the buccal mucosa, under the tongue, or on the palate. Riboflavin deficiency, also called as arabinoflavinosis, leads to glossitis, magenta discoloration, and atrophy of the papillae. Niacin deficiency, called as pellagra, leads to glossitis, stomatitis, and they are the earliest signs of niacin deficiency. Necrotizing ulcerative gingivitis seen in areas of local irritation. B2, B3, B6, and B12 deficiencies lead to hemorrhagic gingivitis and periodontitis. Folic acid and periodontal disease, the deficiency leads to growth problems, loss of appetite, mouth sores, migraine headaches, fatigue, gray hair, tongue swelling, and irritability. Their absence is seen in the oral manifestations as absence of keratinization of the gingival surface, decreased cell turnover rate, diminished resistance to infections, destruction of gingival and periodontal tissues without inflammation. Vitamin B12 and periodontal disease, the deficiency in the body can lead to lower mobility, glossitis, mouth ulcers, pale and jaundiced skin, dizziness, mood changes, high temperature, sensations of pins and needles, fatigue, and disturbed vision. The oral manifestation can be seen as impaired wound healing, worsening of the periodontal status, increased attachment loss in spite of therapy. So, periodontal manifestations of vitamin B12 deficiency, a case study done by Matthews et al. In 2017 linked vitamin B12 deficiency and acute necrotizing ulcerative gingivitis and increased clinical attachment loss. P. gingivalis uses it for metabolic flexibility, so low level of B12 in periodontitis patients is seen. Vitamin B and periodontal therapy, we've seen that Nivea et al. Effects of vitamin B cocktail, that is vitamin B12, folate, thiamine hydrochloride, riboflavin, niacinamide, D-calcium pantothenate, pyridoxine hydrochloride, and D-biotin on periodontal wound healing after periodontal surgery showed improved clinical attachment level compared with patients with received a placebo. The drawback is it is still not clear whether one active component within the vitamin B complex mixture or a combination of different micronutrients cause this effect. Vitamin C. The dietary recommended allowances is as seen in the chart, helps in converting fat into energy, supports immune system as an antioxidant, healthy nervous system functions, and helps in synthesis of collagen. Its symptoms and deficiency in our body is seen as digestive disorders, immune impairment, joint pain, dry skin, dry splitting hair, slow wound healing, gingivitis, easy bruising, and nosebleeds. His oral manifestations is seen as irregular dentin formation, scorbutic gingivitis, and bleeding gums. Vitamin C has a relationship with periodontal pathology has been described historically. In the 18th century, during maritime trading and exploratory seafaring, sailors suffered from scurvy, a vitamin C deficiency disease associated with gingival bleeding and tooth mobility, clinically called as scurvy, is diagnosed when serum ascorbate levels falls below 11 μ. Recent consensus report, the European Federation concluded that vitamin C plays a role in the onset and progression of periodontal diseases, as reported by Chappell IL in 2017. A longitudinal study by Ed Stoddard in 2005 linked the consumption of vitamin C rich grapefruit to improved plasma concentrations of vitamin C and decreased sulcus bleeding scores. Intradermal and subepithelial vitamin C injections used as an adjunct with surgical and non-surgical treatment of periodontal disease led to satisfactory results as reported by Raghavendra et al. In 2018. The literature suggests that the patients with a lower dietary intake or lower blood levels of vitamin C showed a greater progression of periodontal disease than the controls. The intervention using vitamin C administration improved the gingival bleeding in gingivitis, but not in periodontitis. Alveolar bone absorption was also not improved. The present systematic review suggests that vitamin C contributes to a reduced risk of periodontal disease. Minerals. We'll find out the identifying functions and metabolism, the symptoms in case of deficiency, and the sources from which it can be obtained. The mineral nutrients are elements other than the oxygen, carbon, nitrogen, and hydrogen required by the organisms to survive and function normally. Some of the important functions that these minerals perform are boron. They are important in bone retention, found in fruits, leafy vegetables, nuts, legumes, and beans. Calcium, essential for growth and structural integrity of the bones and teeth. Nerve conduction, muscle contraction, relaxation, found in yogurt, milk, cheese, tofu, fortified juices, green leafy vegetables. Chromium participates in CHO and fat metabolism. Muscle function increases effectiveness of insulin, found in whole grains, cheese, and yeast. Copper, essential for red blood cell production, pigmentation, and bone health, found in nuts, liver, lobster, cereals, legumes, dried fruit. Iron, essential for production of hemoglobin in red blood cells and myoglobin in skeletal muscles and enzymes that participate in metabolism, found in liver, clams, oatmeal, farina, fortified cereals, soya beans, apricot, green leafy vegetables. Magnesium, essential for nerve impulse conduction, muscle contraction and relaxation, and enzyme activation, found in whole grains, artichoke, beans, leafy vegetables, fish, nut, and grains. Manganese, essential for formation and integrity of connective tissue and bone, sex hormones, production, and cell function, found in nuts, legumes, and whole grains. Phosphorus, essential for metabolism and bone development, involved in most biochemical reactions in the body, found in fish, milk, meats, poultry, legumes. Potassium, essential for nerve impulse conduction, fluid balance, and for normal heart function, is found in squash, potatoes, beans, fresh fruits like bananas, oranges, and vegetables like tomatoes. Selenium is an antioxidant, works with vitamin E to reduce the oxidation damage to tissues, found in meats, seafood, and cereals. Sodium, essential for nerve impulse conduction, muscle contraction, fluid balance, and found in table salt, canned, and processed foods. Other minerals like calcium, the deficiency is not determined as yet, but it is required for the formation of teeth and bones. Supplementation improves outcomes of non-surgical periodontal therapy. Local application enhances osseointegration. Found in milk products, eggs, canned bony leafy vegetables, fish, nuts, and seeds. Magnesium required for cell metabolism and bone formation. Supplementation may improve outcomes of non-surgical periodontal therapy, found in cocoa, soya beans, nuts, spinach, marine vegetables, and tomatoes. Iron Possible antioxidant effect on periodontium found in red meat, tuna, dry beans, spinach. Zinc for possible antioxidant effect on periodontium, reduces severity of diabetes, reduced periodontitis, found in protein-rich food, spinach and grains. Fluoride, used in supplementation and topical application, prevents dental caries, found in grapefruit, cocoa, tea, dried fruits, nuts, fluoridation, water fluoridation. Minerals in periodontal disease, deficiency of dietary calcium may impact periodontal health as well. Co-supplementation of calcium and vitamin D is commonly used and has a positive effect on outcomes of periodontal therapy, as mentioned by Gracia et al. In 2011. The impact of dietary magnesium on periodontal health is still unclear. So far, only one study has suggested a positive effect of magnesium-rich diets on non-surgical periodontal therapy, as stated by Stoddard et al. In 2012. The study indicates that iron deficiency anemia leads to a reduction in antioxidant enzymes, leading to an increased oxidative stress and worsening of the periodontal disease, as stated by Chakraborty in 2014. A systematic review has further supported the importance of zinc in preventing diabetes-related periodontitis by exerting an antioxidant effect, as given by Pushparani in 2014. Nutritional deficiency and the periodontium. Why does the deficiency occur? The causes of nutrition deficiency could be systemic disorders, increased requirements, loss, drugs, and malabsorption. Nutrition and food affect the periodontal health in 3 levels: contributing to microbial growth in gingival crevice, affecting the immunological response to bacterial antigen, assisting in the repair of connective tissue at the local site after injury from plaque, calculus, and so forth. Nutrition and periodontal disease may influence pathogenesis of the periodontal disease by affecting colonization and invasion of the periodontal microbes, various parameters of host defense system, and repair process that follows periods of active destruction, as given by Enbonwo in 1994. Nutrition is a key modifiable factor for chronic diseases such as periodontal disease, as given by Mariton in 2021. According to Bod in 2003, nutrition affects the immune system, thus it affects the periodontal disease and its treatment. If you see this chart, this is a microbial invasion. Whenever a microbial invasion occurs, there is an increase in the PMN, which release free radicals via respiratory burst mechanism, which in turn attacks the cells and cause destruction. However, when enough amount of antioxidants are present, the actions of these oxidants are nullified. These antioxidants are obtained from vitamins and minerals. The action of antioxidants is preventive, scavenging, and repair, and de novo cell formation. Hence, a perfect balance between the oxidants and the antioxidants are required for a perfect health to be ensured. Literature says that among all the analyzed vitamins, those with antioxidants capacity and effects of immune systems seem to be useful for prevention or improvement of periodontal disease, as well as those implicated in bone metabolism. So nutrition, maintenance care, and periodontal therapy has got something in common. Periodontal health is influenced by a number of factors such as oral health, oral hygiene, genetic and epigenetic factors, systemic health, nutrition. Many studies have observed that a balanced diet has an essential role in maintaining periodontal health. Additionally, the influences of nutritional supplements and dietary components have been known to affect the healing of periodontal surgeries. Studies have attempted to find a correlation between tooth loss, periodontal health, and nutrition. Moreover, bone formation and periodontal regeneration are also affected by numerous vitamins, minerals, and trace elements. So this was just a presentation to critically appraise the currently available data on diet and maintenance of periodontal health and periodontal healing, the effects of nutritional intervention studies to improve the quality of life and well-being of the patient with perineal disease will be most useful to find out how much we can do for our patients in order to help in the healing part. Now, this presentation is brought to you so that you will be able to attempt basic questions that is asked usually in your part 1 papers. So good nutrition creates health in all areas of our existence. All parts are interconnected. Thank you very much for a patient listening. This is a team that I work with at Nair Hospital Dental College. I wish to thank the VISHNU Dental College for having given me this opportunity to make this presentation to you. Thank you very much for your patient listening.
Thank you, ma'am. That was a wonderful presentation from your side. And I think you have one question.
Ma'am, am I audible, ma'am?
Yes, yes. Yeah, tell me.
Ma'am, one of the participants asked, role of vitamin E deficiency in TMJ problems, ma'am.
Vitamin A is supposed to repair blood vessels.
E, ma'am.
E, madam. Vitamin
E. Yeah. Vitamin E is supposed to repair the blood vessels, which gets damaged in most of these TMJ problems, mostly because of the psychosomatic disorders like clenching and grinding the teeth. So those type of repairs are been helped to heal with the help of vitamin E.
Thank you, ma'am. Your wonderful presentation and your knowledge had a significant impact on our delegates, ma'am. Thank you so much. We have a small token of appreciation from certificate from VISHNU ADHYAAN, ma'am. Please accept.
I thought there will be.
Yeah, we'll mail you, ma'am.
Ma'am, you made the presentation so clear that I don't think they had any doubts.
No, I thought they will be asking some questions. What are the effects? What affects the nutrition? So actually, I wanted to tell them, if you have not thought about it, maybe age, sex, body weight, level of activity, and the physiological status of a person who comes to our clinic is a major factor in us looking into nutrition as a dentist. There has been conditions when people, high increase of intake, nutrition also affects us. Low increase also affects us. High would be, say, and if he takes it high, he will have a toxicity of nutrition. And low, and if there is obesity connected with it and no physical activity at all, definitely he'll have most of the problems. And low intake, if you go to see starvation, it will have low body weight, loss of muscles, and many other healing factors will be poorly affected. This is what I thought. They would ask me that how many people have died because of the lack of nutrition and vitamins. Yes, there are reports which says that at least 45% of the childbirth today below the age of 12 died because of nutrition. And if you see in India, there are many people who can starve, or we can take examples of Somalia, Ethiopia and all, where people have died because of a lack of intake, because of starvation. So these all go to the, what is important from our point of view in our exams and our patients that come to our clinic is, they should be well nourished enough to take the insult of our treatment. And we should be in a position to give them good dietary suggestions, which will help them in the healing. That's what I would like to say. Thank you very much.
Exactly, ma'am. What you told in the end was very right and apt also, because the advertisement nowadays, we see that cushy ad in TV. I think even government is taking up all these things very seriously.
Yes.
And I think as working in a government dental college or in a city like Mumbai, wherein you have the most of the population, wherein you can see all sectors of undernourished, overnourished, everything.
Yes.
I think you get to see a lot of cases also with your clinical years of experience and so on. But then most of the patients who come to our private clinics, I think they are well nourished.
Well nourished.
Yeah.
Yeah. I hope I was good enough to give them something for their papers to write down.
Yes, ma'am. Very nice. It was very nice. I think perio can read always the 8th edition of Carranza, wherein nutrition and periodontal health deficiency.
Right.
Very clear. And I think it will be helpful for other departments. Thank you so much, ma'am. It was very nice. It was a very
Thank you, God. Thank you for this opportunity. Thank you.
Thank you so much.
Thank you, ma'am. Thank you so much. You enlightened us. Thank you, ma'am. Good afternoon, dear delegates. We have a lecture from Dr. Darpan, sir. Sir, am I audible?
Yes, yes, you are audible very well.
Good afternoon, sir. The session
Good
is all yours.
Is the screen visible?
Sir, it's visible, sir. You're trying to play a video. It's visible, sir.
Is the screen visible?
Visible, sir.
Okay. Thank you for yet again getting me back on the platform. And I think only the video towards the end of the lecture was a problem. So I think instead of repeating the entire presentation and not spoiling time for everyone, we'll just go back and revise the anatomy part of it once again, and I think that should suffice what was missing from the first lecture for this program. So thank you very much for getting me back again, and thank you for all your requests that we would have this again. I think the voice was not clear at that instance. But now again, I would say that when I repeat this lecture and I go through the flow of the anatomy, it's a pre-recorded video, so we cannot go slow or skip any point. So we'll be going step by step through the video. So you'll have to keep pace with me throughout. So please try and keep the pace with me. Concentrate on what is happening on the screen, and let's go ahead with this. So we'll just revise on the cadaveric anatomy which we spoke about. So you have the condylar head, which is lined by a fibrocartilaginous layer. You see that the head shape is not uniform. It is more of a dome, and anterolateral and posterior it has variable shape. You have the glenoid fossa, which is part of the temporal bone, and you have this also lining with your fibrocartilage, and you have the articular eminence anterior to the fossa. So this is a part of the temporal bone, and now this entire complex is enclosed in a capsule. Now this capsule is a fibrous capsule, and it encloses the entire contents of the temporomandibular joint. Now, this capsule is laterally augmented with the lateral ligament from the articular eminence till the neck of the condyle on its lateral aspect. And as you remove the capsule, you would see that you have the superior joint space and the inferior joint space, and you have the intervening fibrocartilaginous disc in between. So this disc has its dynamic functions along with the joint function. Now, when you see and look at the disc, it is more of a fibrocartilaginous avascular structure, and this helps in mandibular movements when your joint functions. Now, when we talk about joint function, we already discussed that we have two basic functions, which is pure rotation of the joint and the translation of the joint, which combines together to form the complex function of the temporomandibular joint.
Sir?
Yes, Divya. I think give me 2 minutes, I'll sort this out.
Okay, sir.
Is the screen share visible now?
It's visible, sir.
So once can you play, sir?
Oh, wonderful. So I think because of this Vishnu Adhyaan, my revision of the anatomy is gone repeatedly. I think this is third time we will go ahead with this.
Sir, can you just play one, sir, so that we'll be able to see, sir?
Okay. So we'll go ahead with, again, I'll start with the explanation once more, and we'll go ahead with the anatomy of the temporomandibular joint. And let me confirm once if everything is working fine.
It's playing, sir. Unmute. Sir, can you please unmute,
Go
sir?
ahead with the anatomy once more. And so we discussed that the condylar head is a bony component of the mandible, which is covered with fibrocartilage. It is asymmetrical. It has different dimensions anteroposteriorly and lateromedially. You have the fossa, which is the part of the temporal bone, and this is also lined by fibrocartilage. Anterior to the fossa, you have the articular eminence, which is there, and which is the anterior stop to your mandibular movement. And the entire complex of the joint is enclosed within the joint capsule, which is again a fibrous capsule which encloses the contents of the joint. Now, at the lateral aspect of this capsule, you have enhanced fibrous ligament, that is your lateral ligament of the temporomandibular joint, which is one of the check ligaments starting from the articular eminence till the neck of the condyle. Once you remove the capsule, you would see that you have the superior joint space, and you have the inferior joint space, and you have the intervening disc which functions during the movement of the jaw. Now, once you're clear with all these structures, you would see that the disc is fibrocartilaginous structure and it is avascular, and it also gets involved with the dynamics of the joint movement. That's how the isolated disc would look like. And now we have discussed that the joint as such has dynamic two type of motions. One is pure rotation and second is translation. Usually mandibular movements are a combination of these movements. And then once you have pure rotation, you have the disc which sits at its own place. But once you have translation, you would see that the condylar head along with the disc moves forward during the motion of the mandible. And that's how you achieve reasonable mouth opening, with a combination of rotation and translatory movement of the mandible. So initial few millimeters are by rotation of the mandibular condyle, and further ahead to achieve complete mouth opening, you would have the translation which happens in the mandible. Now let us go on to the muscles which are helpful in this movement. So you have lateral pterygoid muscle attached to the lateral pterygoid plate. This is how it would look like. It runs from medial to posterolateral aspect till the neck of the condyle. Now it is attached to the areas of the sphenoid bone here and to the lateral aspect of the lateral pterygoid plate. Now from here it runs backwards posteriorly to get attached to the neck of the condyle. One, let us look at this specimen from the medial aspect. So this is your lateral pterygoid plate. The muscle is running from the lateral pterygoid plate till the neck of the condyle. At the area of attachment called the pterygoid fovea, which is a depression in the neck of the condyle, and the part of the fibers of this muscle are also attached to the intervening disc. Now the capsule is removed, you see the superior joint space, inferior joint space, and this is the intervening disc. So you have superior joint space, inferior joint space, intervening disc, and that also gets attachment of some of the fibers of the lateral pterygoid muscle. Now let us have the medial pterygoid attached to this. On the medial aspect, you see that medial pterygoid runs from the angle of the mandible till the medial aspect of these pterygoid plates. Now this particular muscle finds its attachments at the medial aspect of the lateral pterygoid plate and the medial aspect of the medial pterygoid plate, and also at the retromolar area or the tuberosity area. Now from there it runs downwards and backwards to get attached to the angle of the mandible on the medial aspect. And then now let us go on to the lateral aspect of the skull and try and learn about temporalis. So this is coronoid, which is attached here. The zygomatic arch is being cut. Now you have a large fan-shaped muscle which is called as temporalis. Now it starts from the area on the lateral aspect of the skull along the temporal lines and covers this anterior lateral aspect of the skull. It runs downwards and narrows down and passes below the zygomatic arch and the zygomatic region to get attached to the lateral aspect of the coronoid of the mandible, anterior coronoid of the mandible, and medial coronoid, and runs downwards, sometimes even till the area where your last molar is present. So you may find some tendons there. Now this entire system of temporalis muscle is enclosed in a fascia. So this is how it would look like when the entire muscle is enclosed in a fascia and goes below your zygomatic arch to get attached to your coronoid process. So I hope that is clear till this region. You see the entire muscle system covered with the temporal fascia over the muscle. Now let us go ahead to learn a little bit about masseter, which is one of the large muscles on the lateral aspect of the face. Now this originates from, again, your areas from the zygomatic arch on the lateral aspect and the body of the zygoma, anterior 2/3, and then posteriorly or medially it is attached to the entire zygomatic bone and the entire length of the zygomatic arch. Now it runs posteriorly behind and goes and attaches again to the angle of the mandible. You have the lateral fibers and you have the medial fibers, which are more vertical. Now this is the area on the lateral aspect of the ramus of the mandible, more of angle and ramus, where this muscle is attached onto the lateral aspect of the mandible. Now this on the medial aspect is continuous with a sling with medial pterygoid muscle, and this sling of attachment of two muscles is called as pterygomasseteric sling. Now mandibular movements are usually controlled by check ligaments. These ligaments control the excessive movement of the mandible. So once you open the mouth, all the muscles are aiding. You have certain ligaments which limit the opening of the mouth. So one is your we have already learned the lateral ligament, capsular ligament, and then you have this stylomandibular ligament from the styloid process to the angle, and you have the sphenomandibular ligament. A stylomandibular is from styloid process till the angle, and your sphenomandibular is from the spine of sphenoid till the lingula at the mandibular foramen region. These are two main check ligaments. Now let us learn about accessory muscles which also aid in the mandibular movement. Let us start to learn about mylohyoid, which forms the floor of the mouth as such. It finds its attachment at the medial aspect of the mandible bilaterally in around the mylohyoid lines, and then both the muscles attach in the midline through a raphe, and posteriorly it finds its attachment to the body of the hyoid bone. Now, this is how it forms the floor of the oral cavity, and this aids in the depression of the mandible during mandibular movement. Now, let us go on and attach geniohyoid muscle to this specimen. And now you would see that it finds the attachment from the genial tubercles on the medial aspect and midline of the mandible, which runs posteriorly to get attached again to the body of the hyoid bone. Now let us attach stylohyoid muscle to the specimen. A stylohyoid is again from the styloid process, runs downwards, and it has an opening on the posterior aspect for the digastric muscle, a split in the muscle. So this is attached to the greater cornu of the hyoid. This is yet another muscle which aids in mandibular movement. Now let us attach digastric muscle. Now, digastric has got two bellies, the posterior belly. Now you would see that the posterior belly enters this slit or the opening in the stylohyoid and proceeds further ahead. And now once you see the origin of this muscle, it is attached at the medial aspect of the mastoid process of the skull. So there how it comes downwards and anteriorly enters the slit in the stylohyoid, comes down, and then you would see that you have a pulley-like structure which is attached to the hyoid bone, which holds the tendon of the anterior and posterior belly of the digastric muscle. Now this is the area of the lesser cornu where your pulley is attached, and then you have the anterior belly of digastric. Anterior belly of digastric, from the area of the tendon where the pulley is there, you have it running anteriorly and getting attached to the digastric fossa, which is present onto the medial aspect of the mandible. Now, with this understanding, also a reminder that two bellies of digastric find different innervation from nerves that you should be knowing about. And that's in gist and summary about the anatomy of the temporomandibular joint. Now, when we learn about all this, we already had learned in the main presentation on the first day, the first presentation, that how does the mandible function and what are the biomechanics and the dynamics of the mandibular function. And then also we did learn about the action of individual muscles and how they help in the mandibular movement and in turn the joint movement, which is the effect of the motor activity of these muscles. So I thank all of you once again for patient listening, and I hope the presentation was clear at this instance. Thank you very much.
Thank you so much, sir, for your repeated efforts to make our delegates understand the TMJ anatomy. You have enlightened us with your knowledge, sir. Unmute, sir, please. Unmute, sir.
Hope we can wind up now.
Yes, sir.
So this time it was, I think there was no confusion, no? It
No,
went
sir.
well.
It was crisp and clear, sir.
Yeah, okay. Thank you very
Thank
much.
you.
Thank
Thank
you.
you so much, sir.
Hi.
Very good afternoon. Very good afternoon, sir.
Yeah. How are you?
I'm great, sir. Thank you very much, sir.
It's really nice, and then we had a very nice time with all the lectures, sir. And yours is excellent 3D view you showed. Actually, all the students wanted actually that. Really, it's nice. Thank you for accepting our invitation. Hope we'll meet in VISHNU Dental College again.
Thank you, sir. Thank you very much for this invite. And in fact, it was my pleasure being here. And it's always good to connect with all of you yet again. This anatomy lecture actually is a lecture from Louisville University from one of the professors who had done an extensive cadaveric dissection on this. He was basically a plastic surgeon, not a maxillofacial surgeon on those days. Now he's passed on this literature to us in the file formats, and it's copyrighted. So although there was a request that we could share it, but I have promised that we will not share his lectures across any platforms. So that's what's the reservation, that we could not share this lecture. But I sincerely thank everyone for patient listening and again connecting to Vishnu yet again. Thank you very much, sir.
Yeah, our director, Dr. Anand, is also there online actually. He wanted to listen to your lecture actually. He's there. Maybe I think he can speak.
Oh, that's my pleasure, sir. That's my pleasure.
Dr. Anand.
Good afternoon, Dr. Darpan.
Very good afternoon, sir. Very good afternoon, sir.
It was an excellent, I mean, we have always learned anatomy, but visualization was always a part. I mean, though we have been into dentistry for the last 20, 30 years, in spite of that, I think visualizing the anatomy in a three-dimensional has always been a challenge.
Yes,
And,
sir, exactly.
The video by the great professor, I think has really opened all our minds. And I think now it is much more easier to understand the various attachments and how actually these muscles play a role in mandibular movement. Thank you once again for presenting a beautiful video and your presentation.
Thank you, sir. Thanks a lot for this opportunity. Pleasure is all mine.
Thank you, sir. Please retain for the valedictory function, sir. We'll shortly start.
I am there. I am there.
So now we have come to an end of the Vishnu Adhyaan 2022. So I thank all the speakers of today, Dr. Swagatika ma'am, Anil Babu sir, Veeramal sir, Vinita ma'am, Shaila ma'am, Mala Dikshit ma'am and Darpan sir for enlightening us with your nice presentation, taking time from your busy schedule to be the guest speaker at our Vishnu Adhyaan. Your presence and wise words helped us to magnify our cause in the best possible way. All thanks to your enlightening words that inspired so many people out there. Thank you. I hand over the session to Dr. Anand sir. Dr. Anand sir, Associate Professor from Department of Oral Medicine and Radiology. The session is all yours, sir.
Thank you, ma'am. Before proceeding to the special occasion to celebrate birth anniversary of founder chairman of Sri Vishnu Educational Society. Late Dr. Baby Raju sir was the founder chairman of Dr. Baby Raju Foundation, Sri Vishnu Educational Society and Vishnu Dental College. He was a prodigy of industrial discipline, unusual perception, scholarship, workmanship, and creativity. He was a man of vision, inspiration, confidence, diligence, and above all, humanity. His achievements in the immeasurable services in the industrial field as well as towards the society won him the Padma Shri in the year 1977, and Padma Bhushan was one of the most prestigious awards by the Government of India in the year 2001. He was also awarded honorary doctorate by JNTU, Hyderabad. Being an earnest, scholarly man, he knew the best value of education and thus diverted his focus from industrial field to the vast field of education to set up a series of colleges under Baby Raju Foundation and Sri Vishnu Educational Society with 2 campuses in Narsapur and Bhimavaram respectively. And now we'll start with the valedictory function. I, Dr. Anand, from Department of Oral Medicine, take this opportunity to welcome each one of you to the valedictory event of Vishnu Adhyaan, a national level rapid revision program for postgraduate students. This is an immensely exciting evening for our institute, Vishnu Dental College. The actively growing participation of students from different colleges every year affirms the relevance and meaningfulness of this program. All in all, it was a knowledgeable event. I hope you have gained much needed information with good interactive sessions. Thank you all. Now we'd be very happy to display responses from our guest speakers. And I'm reading one of the response from Dr. Vandana ma'am. Vishnu's platform, a touch of academic festivity and a bonus of learners. Each one of your involvement and contribution is a service to Ma Saraswati. And we got the opportunity to share this platform, Wonders of Wound Healing. Thank you all. It's a shared blessing that we have one of the best of country's management under the able leadership of Sri Vishnu Garu. My pranams to him. This is the message we have received from Dr. Desai Vandana ma'am, HOD of Periodontics. Now I request Director of VISHNU Dental College, Dr. M.A. K.B. Raju sir, to address the gathering on this occasion.
A very good afternoon. Thank you, Anand. I am also known as Dr. Anand. I'm Anand Raju, and so there's always a confusion in the college. Anyway, thanks a lot, Anand. And I think at the behest, I have to thank from the bottom of my heart to all the resource people who have spent their time and the urge to share their knowledge with the students. I think being a teacher is probably the noblest profession I would consider, because this not only involves in imparting education to the individuals, but also a lot of other things. And as students, and all of us have had great teachers in our lives who made a big impact in our lives. So I would like to thank from the bottom of my heart to all these teachers who have spent their time, and especially Dr. Darpan, as he rightly said, it was his third time repeating the anatomy of TMJ. I guess no one can better write an exam on TMJ anatomy after listening to his lecture. And I would also congratulate the team VISHNU ADHYAAN, which is hosted by the three departments of VISHNU Dental College, the Oral Medicine and Radiology, the Oral Pathology Department, and the Oral and Maxillofacial Department. All the faculty who have been involved in organizing this wonderful event, I would like to thank you from my bottom of the heart. And the least, but not the least, I would say, the students who have actively participated in this program. All of you know that it's a very important program for you, because I guess probably in the next couple of weeks, all of you will be facing exams. I would congratulate and also probably give you my best wishes for your exam. Be confident, and I'm sure you will do very well. And I think probably very close to the future, I think we have Deepavali coming up. So I would wish every one of you a happy Deepavali. Thank you once again.
Thank you, sir, for your best wishes for the success of this program, VISHNU ADHYAAN 2022. Now I request Professor Ann Head, Associate Dean of Academics, Dr. Sudhakar Reddy sir, to propose vote of thanks on this occasion.
Thank you, Anand. A delightful evening to everyone present here. On behalf of all, I deem it a great honor and privilege to stand before you to say words of gratitude for being with us all the 3 days of VISHNU ADHYAAN 2022. With deep reverence and gratitude, we thank our valued Chairman Sir, Vice Chairman Sir, Secretary Sir for their constant strive to make things better and improve towards progress. I also thank our Dynamic Director, M.A. K.V. Raju, Principal Dr. Suresh Sajjan, Vice Principal Dr. A.V. Ramaraju, and Associate Deans, Head of the Departments, Faculty of VDC, without whom this program would not be possible. We sincerely thank all the invited guest speakers for their enormous cooperation, devoting their valuable time, sharing their experiences and knowledge in this platform, VISHNU ADHYAAN. I would like to extend my heartfelt appreciations to student delegates, especially for their active participation to make this event a grand success. Hope all the lecture series had benefited to you people, and I wish all the very best for your forthcoming university examinations. I also thank our organizing team, scientific team, treasurers, registration team, technical team, especially Dr. Vineet, and postgraduate students of VISHNU Dental College, who have been a great support for organizing the event. Last but not the least, we also thank General Administration, our Administrative Officer, Mr. Varmagaru, for their constant support. Thank you one and all. Jai Hind.
Now I request everyone to stand in your places for National Anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता। पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा। विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा। तव शुभ नामे जागे तव शुभ आशीष मांगे। गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता। जय हे जय हे जय हे जय 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 हे।
Thank you, one and all.
Jai Hind.